Okay, good morning to everybody. Welcome to the third day's sessions. Today happens to be the so-called National Mathematics Day, which is the birthday of somebody called Srinivasa Ramanujan, one of the mathematical considered to be a genius. So this is celebrated as National Mathematics Day, and welcome to enjoy our sessions. Today we have 5 talks, and the last talk is by the director of this institute. I see that people generally slowly come just after the session starts. Please don't do that. I think it's kind of disturbing. So some of the speakers are very sensitive about that. Since I've been told, that's why I'm announcing it. Please come beforehand, just 2 minutes before. Okay, so for the first session, I welcome Professor Sudipta Thung to give a talk, and I also welcome Professor Sindhu Radhakrishna to chair the session.
Thank you, Suri. Good morning, everyone, and welcome to this morning's session. We begin with a bang with a talk on evolutionary biology. Our speaker for the day is Dr. Sudipta Thung. Dr. Sudipta finished his PhD from IISER Pune and then went on to do a postdoctoral fellowship at Harvard University. He is currently a welcome DVT fellow at Ashoka University. He works on the evolutionary and ecological dynamics of organismal and suborganismal levels, and he'll talk to us more about it today. Thank you, Sudipta.
So you can count the number of animals there.
Thanks, Sindhu. I hope I am audible from the back. Great. Good morning. So let's start today's talk. Although today is the mathematics day, we will start with a biology talk, and we will see that mathematics is kind of the natural language of biology. We'll see if we can connect. So what is biology? Biology is a study of life. And what is the beauty of life? Is its diversity, isn't it? So how did we get such a rich diversity? So that's the idea. Okay. So let us hear this video.
Our planet has millions of species, over 300,000 beetles alone, 17,000 butterflies, thousands of mammals, fish, and birds, all astonishingly different. How did so many species come to be?
In fact, that's a real question. How did we get such rich diversity? Isn't it? So before we go to the topic, can you tell me exactly what are you watching here? What are you seeing? Dogs and puppies. Okay. Anything else from the terms of genetics, maybe? Sorry? Okay. Well, I have just given you this picture. I have not said anything. You may have already assumed certain things, the two assumptions being maybe these are the parents and these are the pups. Okay? It's important to state the assumptions. If we have that assumption, we can also see certain patterns. What pattern do we see? Do they look exactly identical to the parents? No. Do they look exactly identical to themselves? No. But they do look like dogs, right? The pups. Okay. So the two common things what we can get out of this picture is probably that the offspring, they resemble their parents, but they are not identical neither to themselves nor to the parents. And that is the crux of this origin of this magnificent biodiversity. We'll slowly build upon it. So there are 3 things which are key in the context of understanding this source of diversity. The 3 things being variation, selection, and heritability. You have fair idea, rough idea regarding the 3 things. Variation, that is not everybody is identical. Selection is some process by which some selection is happening, certain features are being selected. Okay. We'll elaborate on that. And heritability, what is heritability? That is, the traits can be passed on to the next generation, isn't it? And do you know what these three things in together are called? Sorry? Say it loud. Natural selection? Natural selection, just this part. Evolution, exactly. So basically together we call them as the process of evolution. We'll essentially elaborate on them. Each of them, heritability will not touch as such, but we can discuss during the coffee session exactly how it may impact. Okay? Okay, so variation. How do we get such variations? So basically, variation is the source or raw material for evolution to happen, right? So how do we get variation in organisms? So raw material of evolution. Crossing over, I see. When do we get crossing over? During mitosis or meiosis? Meiosis, correct? So that's the first thing, origins of variation. One is of course before, so this is what you are probably talking about, which is the recombination. But even before that, we can have something called mutations that can create variation in DNA sequences. And that is very relevant of many things, this antibiotic resistance. You heard about it? Right? This recombination is one of the major driver of variation in organisms where recombination can happen, say higher eukaryotes, right? Is there any other source of variation? Anybody knows of anything? There is a peculiar feature in some of the cells where some of the genes, they jump around in the genome. Have you heard about those? Transposons, right? It also creates immense variations. Okay, all right. So these are the different kinds of sources of variation. Now suppose we have the variation built in the population. So what is a population, by the way? It's a collection of individuals, right, which are cohabiting at one place and interacting with each other with respect to nutrition and reproduction. That's the simplest definition. Okay. Now in that population, we are saying that variation exists. That means not all are identical. So let us see how selection may act on that. Suppose we are considering this is like a cartoon diagram. A bunch of beetles are there where variation exists. Variation with respect to coat color, let's say. So a bunch of green beetles, and in that some of the orange beetles are also there. So therefore, not all are identical. So therefore, variation exists. Okay. How selection can act? Now suppose I introduce another actor in the whole play, that is this crow. They like to eat the green ones because they are very prominently visible and they can eat them. Okay, that's it. So variation, and introduce one agent who can select. Okay. Now with these two combination, it becomes like very interesting. So what will happen in the next generation and subsequent next generations? Interesting. So first of all, they will quote unquote select out the green ones slowly, okay, because they are eaten up. So they will obviously, those which are eaten up, they will not be able to contribute in the next generation. Automatically the contribution, per capita contribution of these orange ones will increase in the next generation, okay. So automatically you will get little more number of orange ones, and if you iterate the process again and again and again, you will eventually probably will have a bunch of orange beetles eventually, isn't it? This is essentially the process of natural selection. Okay. Here the actor is the natural agent that is essentially selecting. We can potentially do this selection by ourselves also. In fact, I'll come to that example, that where human can select certain traits, let's say different kinds of puppies. We can select certain traits and then we end up having a particular kind of organism. So that is called selective breeding. Okay. So in terms of natural selection, there are various different examples. One can sort of think evolution being like a thing of past, but it is actually happening all the time. This natural selection is acting all the time with us. One of the canonical poster example of this kind is of this peppered moth of London. Have you heard about this story? This is one of the most canonical example in textbooks. That's why I just showed the picture but skipping it. Instead, I am giving you another example which is relatively lesser known. We can always go back and discuss about it if anybody is interested during the coffee break. Okay? And this example I will show through the video simply because it's fascinating to see it exactly how it is happening. Okay.
We've come here to the Valley of Fire in New Mexico, a battlefield, to find one of the tiniest soldiers and what it can teach us about how evolution works. On the desert sands, the rock pocket mouse blends in perfectly, its light-colored fur concealing it from predators. But on dark lava, the same fur makes the mouse stand out, attracting the many creatures that see it as food.
These mice are the Snickers bar of the desert. They're eaten by foxes and coyotes and rattlesnakes and certainly by owls and maybe even occasionally hawks. And most of those predators are visual predators.
So what happened to the pocket mice that found themselves on this new terrain? When I accompany biologist Michael Nachman onto the lava, it doesn't take long to find out.
Oh, this one's closed.
Nachman has been collecting mice unharmed in traps. And it's a dark one.
It is, yeah.
Now, are most of the ones you find up here dark?
Almost all of them.
Not only have the mice here evolved to be as dark as the rock, the color change has occurred precisely where it will conceal them from hunters. Now, a bit of a white underbelly, too.
That's right. All of the dark ones here and on other lava flows have a white underbelly, and presumably there's no selection for dark on the belly because predators are coming from above.
Left to themselves, the mice show no preference for light or dark rocks. It's the predators that have made the difference.
The change in color over evolutionary time in the population is driven by predators weeding out the mice that don't match their background.
But how did the dark mice arise in the first place?
When a black mouse appears in a light population of mice, that is usually going to be due to a new mutation. And those are random and rare events.
To fully understand the pocket mouse transformation, Nachman moves from the lava to the lab. He and his team extract DNA from light and dark mice taken from one desert region. The aim? To find one or more genetic mutations that cause dark coloration. A mutation is a change in the chemical letters that make up our genes. It's a copying error that may occur when our cells divide. Mutation seems to mean that something bad has happened. Well, mutations are neither good or bad. Whether they are favored or whether they are rejected or whether they're just neutral depends upon the conditions an organism finds itself. So for the pocket mouse, a mutation that caused the mouse to turn black, that is good if you're living on black rock, and it's bad if you're living out in the sandy desert.
The light mice are all on the bottom, here.
Fur color is a trait controlled by many genes. To figure out how dark mice evolved, Nachman focuses on how these genes differ in dark and light mice. One by one, the genes prove identical, but at last, something does turn up. The difference between dark and light mice boils down to a difference of 4 chemical letters in a gene called MC1R. Because the gene controls the amount of dark pigment in a mouse's hair follicles, a mouse with these mutations grows dark fur, which gives it an advantage on a dark background. But still, that's one mouse. How would its dark fur spread to a whole population?
This lava flow is about 1,000 years old. And so you might wonder, has there been enough time? It's only been 1,000 years. It's a very short period of time for a new mutation to come along and spread so that all of the mice on this lava flow are black, because really they all are.
Indeed, such a rapid spread of a mutation may seem unlikely until you do the math. And the reason is that while only one new mouse born in 100,000 may be black, hundreds of thousands of mice are born in any given year. And then those mice that are black have enough advantage that their babies do better and they have more offspring, and their offspring have more offspring, and just about a 5% advantage compounded year in and year out can very quickly turn the whole population black, as we see today.
Does that make sense? And this is something which we can very nicely model as well, okay? That with slight bit of selective advantage, how with, basically the entire population becomes of one character which is favored by natural selection. Okay. So, in fact, there were several different ways of modeling this principle. One of the particular way is essentially using this framework called Price's equation. Again, this is like starting with a population of number of individuals like NT at Tth generation, and if we consider some trait being Z, which every individual has, you can compute something called like change in average trait value in one generation. And again, if you do that, and if you include something called fitness, fitness is nothing but a composite trait which essentially gives you an idea regarding like how much offspring you will leave behind in the to the next generation, contribute to the next generation. Then what you can get, say just to get a concrete example, suppose we are talking about wing length for some flying creatures and their fertility being the measure of fitness, like how much offspring they are going to leave behind. So, we can compute something like the rate of change or change in average trait value per generation. Okay. And again, the calculation is pretty straightforward. Once you do by hand one by one, you will essentially get something like this interesting, which is the change in average trait value in one generation is 1 by average population fitness and into covariance of the trait and the fitness plus some estimated value, estimates of expected value of the fitness and something called delta Z, which is nothing but transmission infidelity. Like, so basically no transmission of any trait is perfectly 100% complete, right? So, some amount of fidelity infidelity quotient if we can figure out, and that contributes to this particular term. And this particular equation is called Price's equation. We can essentially, using this in a very generic manner, we can track exactly how the traits can potentially change over time. The interpretation wise, this is essentially the covariance. Covariance means nothing like, say for example, we create a graph where Z, that is the trait value, if we increase the trait value and if the fitness also increases, then we say that they have positive covariance, correct? So, if we have a positive covariance and if we for the time being consider like the trait is perfectly heritable just for the time being and ignore this term, we should have a positive change in the trait value. So, that is the whole idea. You can rewrite the equation in this way that this is the slope of this Z and W graph. So, basically this covariance can be written as the variance of Z into the regression coefficient of the fitness and the trait value. So, therefore, you can see that if you have more variation, the rate of change of the trait average trait value will be more, which is in some sense like rate of evolution will be more. Also, you can see that if the slope of the fitness and the trait is positive, you will have a positive change in the trait value. If the slope is negative, then you will essentially have negative change. That means the trait will slowly recede from the population. Does that make sense? So, now, so basically this particular portion captures the natural selection, this particular portion captures something called the transmission bias. Depending on how is the transmission infidelity, you can bias this term, and in certain situation they can overwhelm even the natural selection component as well. One of the canonical example of such kind of shift in trait value, which is also often known as something called character displacement. Okay. One of the poster example of this character displacement is seen in these birds. What are they? These are often known as something called Darwinian finches, and the work is done by two giants of evolutionary biology, Peter Grant and Rosemary Grant. Okay. They were studying these finches in Galapagos Island for almost 20 years or so. Okay. How did it begin?
The Galapagos Islands are one of the most spectacular landscapes in the world, home to a variety of species that live nowhere else. Biologists Peter and Rosemary Grant have been seeking answers to how species arise by focusing on one of the smaller islands called Daphne Major.
When we started out, we had no plan for the long term. In fact, we thought it was just going to be just a few years, maybe 2 years.
2 years have turned into a 40-year odyssey. The Grants have returned every summer since 1973.
Oh, that's a bird.
Is that 3 or 6?
That's Trio Metal 6.
Here, they've made some of the most remarkable observations in the history of field research as they studied the famed Galapagos finches. The finches were first brought to scientists' attention by Charles Darwin, when his voyage around South America brought him to this cluster of islands 600 miles from mainland Ecuador. These volcanic islands are geologically young. They began rising from the ocean floor less than 5 million years ago. At first devoid of life, they now support a modest number of species. Among them, 13 species of finches, found in various combinations on the different islands. The birds live in diverse habitats.
The islands are very different from each other. They differ in size, they differ in topography, and in height.
Larger trees grow at higher elevations, while low islands have mostly cactus, grasses, and shrubs. In these diverse habitats, the finches have evolved many ways to survive. So, Rosemary, what's the important difference between these birds?
This little warbler finch with its very fine needle-like beak is perfect for picking off insects. This one is the woodpecker finch with a rather more robust beak. It concentrates on beetle larvae and termite larvae. Then we have the cactus finch with a much longer, sharp-pointed beak, which probes into cactus flowers. And then these three species are the large, medium, and small ground finches. So, Sean, the basic idea is the beaks are tools, and you need the right tool for the right job.
Clear so far? So again, I mean, I cannot take you to the Galapagos Island and actually see those birds, but I can take the help of the video. Okay. So you see, so these are the basic premise. So therefore, there exists variation among the beak depth. And now exactly how natural selection operates, you will see even in the next clippings that a huge amount of drought sort of hit the island. Okay. And therefore, there was no vegetation almost, and that acted like a natural selection, okay, the agent of selection.
Virtually no rain fell for the next 18 months.
The vegetation practically disappeared, apart from a few trees without any leaves, and of course the cactus bushes were still there.
Now the medium ground finches had to compete for scarce food.
They started off with a big food supply of small seeds, medium seeds, large seeds. As these small seeds became very scarce, they had to turn increasingly to the large and hard seeds. Well, only birds with large beaks can crack open these woody, spiny fruits.
Fair enough. So now only the larger seeds are there. So can you guess what will happen? The birds with the smaller beaks will not be able to get secure food. So that is the data collection. And how do you interpret the data? That is what is here.
Well, I'm showing here a distribution of beak depths of the population in 1976. The survivors of this group are shown in black.
Oh, so the larger the beak, the better your chances.
The larger the beak, the higher the likelihood of surviving through the drought of 1977. 18.6 grams.
When they looked at the offspring, they found an even greater surprise. The average beak depth was more than 4% larger than the previous generation. Natural selection had changed the average beak size.
Fair enough? Okay. So now let us ask this question, that does evolution always sort of lead to gain in traits? Like the beak size became more, some kind of traits are gained, like let's say coat color and all these things. Or does it often like some trait is there and it can potentially disappear as well? Interesting. Almost similar kind of example can be found if we look into the caves of Mexico. They are called the blind cave fish. So here inside the cave, water is filled and you find fishes over there. Obviously, the place is pretty dark. What kind of evolution do you expect? In fact, if you see those fishes, it's essentially the same species, okay, which is found outside versus these guys are found inside the cave, and you find no eyes. Why? No use of it, and therefore no point spending energy maintaining it. You may as well spend that energy in something else. In other words, suppose in a population with variation, there is one individual or couple of individuals who can invest that energy in something else, let's say reproduction. They will leave behind more offspring than the ones who are spending more in the eye formation and eye maintenance, isn't it? And eventually, the property that you do not invest in the eye in this situation will essentially, that property will proliferate over time. And generation after generation, you will have some fish which has lost their eyes. Okay. Now, let me come to this particular question, that does human, us, impact evolution? Which we have already said, of course we do, simply because particularly let's say crop generation, we do a lot of selective breeding that is essentially impacting evolution. Not only for that, even for our quote unquote comfort animal, that is let's say dogs, we have created such large variety of breeds through selecting certain particular features and doing that generation after generation. Does that make sense? However, what I was alluding to here is the other kind, where inadvertently does any human action lead to evolution of certain features in organisms. So one of the slightly on the sadder story is related to these elephants. Okay. These are elephants of Mozambique. Do you see any particular feature which strikes you? Interesting. No tusks. Elephants, no tusks. These are essentially the tuskless elephant story. What has happened? Can you guess?
Throughout history, humans have threatened the survival of elephants. Poaching has killed staggering numbers of these powerful mammals. In Mozambique's Gorongosa National Park, the elephant population was nearly wiped out completely. Only a rare natural trait seems to have saved a small number: tusklessness. Yet this evolutionary development could come at a high cost. In central Mozambique, conservationists have been closely observing the elephants of Gorongosa National Park. 50 years ago, around 2,500 of these African giants roamed this area. But from 1977 to 1992, Mozambique was plunged into a civil war, a human conflict that also killed around 90% of Gorongosa's elephants. Thanks to conservation efforts over the past 2 decades, numbers are on the up again. Today, the park is home to approximately 800 elephants. But researchers monitoring them have noticed an unusual trend. Many of the females are lacking tusks. African elephants are occasionally tuskless. It's a trait that occurs naturally in around 2 to 4% of the female population. In Gorongosa, before the war, the proportion was a bit higher than average. 18.5% of the park's female elephants were tuskless. But of those that survived the years of conflict, 51% were tuskless. And 33% of those born after the war were also lacking tusks. According to conservationists and evolutionary biologists, what we are witnessing here is natural selection in action. The impact of human conflict has literally changed how these elephants look.
All right. So basically, it's not just biology, it's like sociology, everything plays in understanding this diversity that we have now, and also our actions matter. All right. So now, with all these questions, we can always say like, okay, is this the only way we can have evolution? In fact, there is one way we can have evolution without natural selection, and that is through essentially something called the random genetic drift, where chance matters. One such poster example of this kind is, or rather schematic would be something like, let's say, from the mainland, one variant just jumped and happened to come and colonize a particular island purely because of the chance. Okay? And therefore, the genetic composition of this island will be very different from the mainland. And this is basically something called founder effect, which is one poster example of something called the random genetic drift. Do we have such example? We do. Say, for example, for that, we need to look into something called the Pingalap Island, the island of color blindness. What happened in that island? The island is located almost like nowhere in between the sea. Over there, in 1775, one typhoon hit and only 20 survivors were there. One of the men carried a rare gene which leads to total color blindness. This particular gene normally is ultra rare, like very rare to have like total color blindness. So it's almost like 1 in 30,000 globally, but because of this situation, because of this chance event now in Pingalap Island, you see that almost 1 in 10 islanders is completely colorblind. So what changed? There is no what changed this genetic composition of these Pingalap Islands? Essentially, it's the chance event. So we can say like evolution isn't always about survival of the quote unquote the most fit individual, fit as in matching the environmental need. So that kind of fit of the fittest, but sometimes it is almost like survival of the luckiest as well, isn't it? Having said that, all these things are happening in natural situation. Can we study all these things in the lab in a very targeted manner? If not anything, at least like in a proof of principle manner. With actually that is exactly what we do in our lab in Ashoka, like many other places in India and outside. This is something called the tool of experimental evolution. And as a sort of environmental forcing, we are interested in the impact of something we love, which is food, right? So food sustains life, but we also know that food is far more than just being the fuel to sustain our life. What we eat also matters, right? So as a country, if we look at India, exactly what are we eating? So this is one study which shows that of our daily energy intake, almost 62% we are getting the energy from the carbohydrate, and about 12% is from the protein. And among carbohydrate, what exactly are we eating? This is like a state-wise survey, and it's primarily rice and wheat. They are the dominant ones, with certain bit of mix in millets and all as well. These are all C3 crops, right? Now, given this distribution, 62% carbohydrate, is it that much we need actually? In fact, the same study shows that if we would have shifted only 5% of the energy coming from carbohydrate to proteins, that would have lead to reduce the risk of new type 2 diabetes and pre-diabetics by almost 6 to 10%, which essentially says that this is essentially a situation of macronutrient imbalance. Agreed? Now, given this, there is another study which shows that environmental rise in CO2 can potentially lead to further reduction in protein content in the crops, particularly C3 crops, particularly in rice and wheat. So, what will these two things lead to? This is because of the environmental forcing. So, therefore, we do not have any control over it independent of our habit or anything. What we are eating, if that is becoming even less in terms of protein, that will just exacerbate this macronutrient imbalance, right? That is likely to worsen the macronutrient imbalance. Now, if we look into a completely different part of the world, we see in Kodiak brown bears of Alaska, what is happening? They have two major sources of food. One is sockeye salmon and another is red elderberry. This sockeye almond is protein rich food and this is carbohydrate rich food. Typically, after the spawning season of salmon is over, these red elderberries, they ripen. So, basically the brown bears, they have no conflict as such. They eat first this ones, after the season is over, then they shift for the red elderberries. So, they have almost like breakfast and lunch. But because of global warming and rise in spring temperature, what happened? These guys, that is red elderberries, started ripening slightly early and thereby overlapping with the season of salmon spawning. Now, they have two of their favorite food simultaneously. What do they choose? They happen to be, again based on observation, it happened to be they choose more this red elderberries. It has various different consequences otherwise, but the particular fact what or the particular aspect what we are interested in, that the total diet composition, if you see, it has shifted from shifted towards more carbohydrate rich diet now. How would that have impacted their other features, like say for example organismal performance? So, what are the consequences of altered dietary macronutrient composition? That had been the goal of our laboratory studies. In this particular context, a lot of studies have happened where people have studied this macronutrient imbalance, but they have a major confound with respect to caloric content of the food. So, we wanted to avoid that because we know caloric content has a major impact on organismal performance. So, we essentially kept ourselves limited towards the isocaloric regimes. We kept the food isocaloric and changed the macronutrient balance, being high carb diet versus high protein diet, keeping the food isocaloric. So, you need a model system, and fruit flies happen to be one of the very useful model systems, simply because they are very small and short lifespan, so that you can do multi-generational experiments. You can harbor them in large caged populations like this. This has almost like 2,500 individuals, both males and females. So, therefore, you can generate sufficient standing genetic variation, so that evolution can act on and you can study these. And also in terms of diet, precise dietary manipulation is possible, so that we can give exactly what kind of food we want. And also various aspects of nutrition absorption, storage, metabolism and sensing, they have, the fruit flies, they have commonalities or very uncanny similarities between mammals and us. So, therefore, the results can potentially be translatable as well. So, what do we do? So, we started with first creating the diets which are isocaloric and but vary with respect to macronutrient composition. Starting with this is the normal food, where we maintain the protein is to carbohydrate ratio 2.4. So, this is essentially the calculated baseline media ratio based on one established literature. So, we essentially took the recipe, computed this ratio, and keeping this in mind and the caloric content in mind, Akhila created various different kinds of diets by increasing the protein content or decreasing the protein content and thereby increasing the carbohydrate content of the food. And you essentially measure various different things like, let us say, fertility, pre-adult developmental time, lifespan, and essentially also you measure something like pre-adult viability and wing length. And very interestingly, what you see is something like on most of the traits which are like relevant for evolutionary outcome, you see a major impact. But you see that their viability and body size remains more or less the same, which essentially means the diet regimes, all the most carbohydrate rich and most protein rich ones, they are not like harmful, they are not like poison. If you have anything you put too much, it can potentially act like a poison and malnutritive. But we do not hit that zone. They are not malnutritive, but has impact in important life history traits, the features. So we essentially considered this particular ratio 0.25 being the carbohydrate rich diet, and the corresponding isocaloric protein rich diet component is the other side. And we essentially maintain the organisms in these diets for multiple generations. Now, in this context, also one needs to appreciate that in terms of diet, it also matters when you eat this diet, right? Simply because the purpose of food changes across life. Broadly, we can divide the lifespan into two phases after birth. The first phase is the developmental phase, when we primarily grow, and therefore the need for diet or food is to sustain the growth. Post that, when the reproduction starts, then the main purpose of food is primarily to sustain the reproduction, right, and body maintenance. So what we could do potentially, we can change this diet composition in the two stages, both carbohydrate rich and protein rich, and so we can combine in this kind of full factorial manner that carbohydrate rich developmental diet combined with carbohydrate rich adult or protein rich adult diet, and vice versa, protein rich with the two carbohydrate rich. So that gives us, and keeping the food isocaloric, so that gives us a very interesting kind of full factorial research question, that how diet composition and life stage interacts, that is what you eat and when you eat, how that impacts physiology, life history and behavior, and that we can do in both, we can see this impact both in single generation and multi-generational manner. So here I will just focus on the multi-generation where you see the evolution. So you, from the base population, you can create such kind of protein rich and carbohydrate rich diet and combinations, all the four combinations, you run them or maintain them on these diets for multiple generations. We currently have almost covered more than 100 generations or so, and for each regime we have 4 independent population level replicates, such that we can establish the repeatability of the evolution, right, because the creation of variation can be random, and therefore the outcome can often thought to be random. So to show the repeatability, we have 4 population level replicates, and then what you do, before you do the assay, you maintain them on common garden or rather same environment, such that you do not see the immediate effect of the environment, but you see the evolutionary forcing what has happened during the course of evolution, and you would essentially test them. So what we see is that, again, cutting the long story short, what we see, I'll just go to the final thing. We see that carbohydrate rich developmental diet, irrespective of their adult diet, essentially leads to faster pre-adult development, higher early life fertility, no lifetime fertility cost as such, and also no longevity cost. So carbohydrate rich developmental diet is leading to this irrespective of whatever they were eating during their adult. On a similar note, protein rich adult diet, irrespective of what they were eating during their developmental phase, that is their larval phase, we see almost the same set of traits being evolved over time, except for longevity cost is seen only in case of females. Now that is very interesting. You see, they differ in terms of the composition and also in terms of when they were getting, but still they seem to have similar kind of evolutionary outcome. Now the question which is bothering us is like exactly how? Is it like they are reaching to the same end point through different routes or through same route? Exactly what is happening? How it affects the internal physiology and make a sort of metabolism such that we get to see such kind of outcome. So that is precisely what we are at this stage now. Of course, when you do not have answers, you essentially go back and sit with your notebook and pen, start to jot down what all things you know about how those diets actually affect both physiologically and at the biochemically inside our body, and you essentially start creating something like mechanistic models. You essentially try to see exactly how the feeding and nutrient intake, energy metabolism, growth regulations, and storage allocations, all these things essentially interact to give out the phenotypic output. What is the current knowledge is that, and try to see whether this particular perturbation is fitting or not. So, that is exactly at the step what we are currently. We are also scanning over a large variety of different traits. These particular organisms are, I mean, after evolution, what all traits have been changed that we are scanning, that is we calling like deep phenotyping. And also the idea is to connect it to this rich phenotypic data with various different kinds of sub-organismal data like metabolomic data, proteomic and transcriptomic data, and also what are the genetic changes happening, can we connect it to it. So, that is the whole idea currently. So, that is what we are currently interested in. So, this I will skip a bit. So, not only microorganism, also in macroorganism like Drosophila, in microorganism also you can potentially study these things. Say for example, in IISER Pune, in Professor Sutitde's lab, this Selvestri was studying something called the evolution of antibiotic resistance. Okay. Well, actually she was not studying evolution of antibiotic resistance, she was studying how exposure to UV can potentially lead the course of evolution depending on when you shine the UV light. Okay. So, they were finding very interestingly, over time if you put sublethal dosage of UV depending on when during this exponential phase or at the lag phase you are shining the UV light, they evolve slightly differently, and compared to this process of evolution like the basically UV resistance, they also develop independently antibiotic resistance. During the course of an experiment, nowhere antibiotic was used, but eventually you led to evolution of antibiotic resistance. So, this is very interesting simply because UV light is used for sterilization purposes for ever, right, or rather basically in hospitals and all pretty abundantly, but that itself can potentially lead to such kind of unintentional consequences. So, these are very useful simply because you see that it is not about only us, it is also about essentially the life we have in the earth, the animal health, environment, and of course, intertwined with human health. They are all coming together, which essentially shapes exactly what we are, and also we shape basically what is there in the environment, which is essentially coming together in this single One Health kind of framework, that basically this is like the health of the entire biodiversity which we are talking about, and we should be mindful of them, and we should be mindful of our action on and their consequences on them as well. Okay. All right, that is all I had to share today, and this is my lab currently at Ashoka, and I will be happy to answer any questions or any queries if there is any, and thank you for your patient listening.
Please raise your hands. Sure.
Good morning, sir. I really enjoyed your talk. I had a question that while showing your results, you said high early life fertility. Sir, how exactly do you measure that? Like,
right. So, for that experiment, what we did, it's actually quite painstaking, which actually the way I showed it doesn't capture the thing. So, for each of those 4 replicate population, for each of the 4 experimental regimes, so that is we are talking about 16 population, for each day we have measured fertility of the organisms. There were multiple replicates. So, it's a humongous task, like throughout their life we measured how much offspring they are leaving behind. Basically what you do, you set up mating pairs, 2 males, 2 females in a container, small container, and you let them lay eggs, or basically don't do anything to them for 24 hours, and then flip them to a new container in the next day. So, and then you preserve those vials, the containers which has food already. So, how much ever eggs are laid, they will grow and become offspring, and you count those offspring, and you do that for every day. So, you get lifetime fertility, and over there you will see what is their early life fertility, what is the late life fertility and everything, and there we see only early life fertility is different. not anything else. Not even early life fertility is affecting the total fertility also.
Sir, you also mentioned that they have a faster developmental time.
Correct.
So, sir, could it be related that the fertility is less because they are developing faster? Like
It's the other
more
way.
energy is required in developing faster.
Absolutely. So basically, that is the thought, correct? But if you see the result, you see those who are developing fast, they are also producing more offspring. That is what is confusing us. You are absolutely correct, but this is the evidence. So we need to now figure out what is going on inside that can enable this.
Okay. Thank you, sir.
Any more questions?
Hello, sir.
Yes.
Such an excellent talk. It almost covered what I had learned in last semester. I am from ISA Pune, and
Yes.
Sudhir Dev was my professor.
Right.
So I have a series of questions. So you told that the blind fish had evolved. So how did the fishes with eyes, which had a vision, disappear?
So basically, the way these studies are done is like you do cross-sectional sampling. So you do sampling today, you do sampling maybe like a couple of years after and something like that. So when they did check at a particular time point, they found that more of these blind fishes are there. So automatically it is understandable if there is no other particular environmental forcing is there. These guys, the ones which had eye, presumably they have invested more in eye formation and eye maintenance, and they may have contributed less in the next generation, and thereby they got selected out.
So we know there are deep sea creatures which have eyes. I don't know the fish exact name, but they have eyes. So why don't we see deep sea creatures without eyes?
First of all, there are certain deep thermal vents organism which do not possess the eyes, and also we do see some kind of gradient which has something like eye patch instead of eye fully formed eyes. So that gradients are also visible. If you see any organism which is living in the deep sea and still has eyes, functional eyes, then we need to see exactly what is retaining the eye, so that we need to then study. That basically needs to be studied in a case specific manner. Yeah.
So another thing was you told about the elephants going almost extinct. So when the human were evolving and we migrated from different continents, we know that almost at the same time the larger fauna went extinct, almost.
Which one? Which one?
The large fauna from large animals, large mammal went extinct. So why didn't we see any evolution, like natural selection against, like which was against humans? Did we see anything like that?
Well, you see, it's not, if you see the phylogenetics of human evolution, we are what we call like Homo sapiens sapiens. This is not the only lineage we had actually. We had many other lineages which gone extinct. So it is not like we are something special and natural selection can't touch us. In fact, it's quite the contrary.
No, my question was like when the humans traveled, migrated from different continents, when they came to new continent, they hunted down the larger mammals. So are there any mammals which were like immune to human hunting? Like
that I don't know. I need to check. Okay. Yeah, thanks.
I have a few more questions. Sorry.
Okay. If anybody, okay, go ahead.
Can we check if others have?
Yeah.
Otherwise, you
Yeah,
can
sorry,
continue.
sorry.
Just a minute. We have a question here, so let's take
So we
that
can
and we'll
discuss
come back to you.
further during the coffee break. Thank you.
Good morning, sir.
Yes.
It was a nice talk, sir.
Thank you.
Actually, I have a question. Why India has high prevalence to type 2 diabetes, but it has lower BMI compared to the Western countries, right? So please, does the curve outweighs the quantity or something like that?
We are trying to still understand this. So this is something called, I mean, nowadays that is what is being more prevalent, which is called lean diabetes. That is there, and we are still trying to figure out exactly what is going on, which is contributing to these things. Because I mean, our body is quite fascinating, equally fascinating as the outside world. The way the this carbohydrate homeostasis is being maintained, It is quite remarkable, although we often blame when it doesn't work. But at which state it doesn't work, that is something of concern. Okay. But what you are raising is a valid point. We still do not have very clear answer regarding exactly the anomaly what you are suggesting. We are still figuring it out.
So one more question.
Yeah.
As you have seen that the eye has been disappeared from the fish in Mexican lake.
Yeah.
Why does the color also changes a little bit? It has been orange color.
Fantastic. Tell me.
Sir, maybe because of the dark environment, they have shifted like that. For fluorescent, maybe.
Not so again. I mean, see, these are the things. This eye is one of the most prominent one what people have fixed. So again, I mean, in the context, we need to see exactly what is happening. Okay, there might be this particular orange coloration I am not aware of, but one can always think this way. There are several molecular mechanisms which are changing to essentially get rid of the eye, and those proteins and those genes can potentially have multiple consequences. Yes. One of the things could be the color. Okay, and as a consequential effect, we can have another evolution. Okay, so that is a possibility, but that's a good observation. We can always dig in and do targeted study to see the genetic changes and if any of them is leading to the coat or their body coloration as well or not.
Okay, sir. Thank you, sir.
Thank you.
One last question from there, and then we'll have to.
Hello, sir. It was a very interesting talk. Like, you might have gauged that. So my question is that you said the Kodiak brown bear of Alaska prefers the red elderberg
berries
for its nutrition. Doesn't that affect the entire food chain, the marine food chain of the salmon?
Absolutely. So that is precisely why you have noticed that when I was telling that particular example, I essentially qualified this way that we are targetedly interested in the macronutrient composition shifts. Okay. However, if you look at the biodiversity wise, there are huge impacts in that way. There is actually some studies which are currently happening on the salmon population dynamics. Because these brown bears, they eat a lot of salmon, they essentially keep a check on their number, and therefore there is no such situation called overcrowding. Yes, exactly. So basically their predator is kind of gone, not interested. Okay, and that has huge impact on their population dynamics. In fact, in some of the situation, it is leading to overcrowding, and which is essentially detrimental to the salmon themselves. So these studies are currently happening, and you can study a particular phenomena in various different ways. This is one of such examples. Thank you. Thank you so much.
We have run out of time, but it's been an exciting session. Thank you all. As a token of our appreciation, we'd like to give this to Dr. Thank you very much.
Thank you.
So thank you, Professor Sindhu, also for sharing this. We have a small break, coffee break. We'll come back.
Thank you. Thank you.
I work on this antibiotic resistance. Very nice lecture. So regarding the color, could it be that this is deep where there is darkness? So even if you have color, you won't be predated.
Correct. So So there is no selection against
the color.
Very possible. Very possible.
Hello everyone. There have been some redundant requests from the students, so I thought that I'll just announce. There are two things here. One, the slides that many of you are interested in having it. We have this program running as a YouTube live streaming, yeah, in which the slides will be projected there and it will be available in the YouTube always. So you can either put Resonance Outreach Program or otherwise we will send the link to all of you. The second thing is that every day we are taking photographs and it will be uploaded in a Google Drive. At the end of the fourth day, all the 4 links and the Google Drive will be shared to all of you. Until then we will not do, because it has to be bits and pieces that we have to send as a mail. So you will get a mail with 2 things. One, the YouTube live streaming link, and the second one is the Google Drive link for the photographs. Yeah, thank you.
So welcome to the post-tea session. Before I introduce the speaker, let me ask you a question, because most of you are from Kerala. Which is the tallest tree in Kerala? Kerala has coconut trees and palms, arecanut trees. Okay, arecanut tree is supposed to be taller than coconut trees. The reason I said is that this next speaker is also an expert arecanut tree climber. Not now, when he was a young boy, okay. The story goes that when he was a student of 10th standard, his family went through a very difficult situation. There was no food on the table, mother was suffering from disease, and then somebody said, if you climb arecanut trees, you can make some money. So he did climb arecanut trees as a 10th standard student, and there was food on the table, and his mother could be brought back to home from hospital. And so he could even jump from one arecanut tree to another tree. Okay. So 35 years from, around 35 years from now, he is Professor Kana Sureshan, a professor of organic chemistry of ISER Thiruvananthapuram, and I would say that he is the most prolific researcher in chemistry in ISER Trivandrum right now. If you look at Google Scholar, this year itself he has published more than 12 research papers in chemistry in the topmost chemistry journals. His area of expertise is in carbohydrate chemistry and supramolecular chemistry. He works on an area called gels and gelators. Okay, these are molecules which form gels. And very beautiful chemistry, and not only of academic importance, but also of applications. There are applications where oil spills can be controlled, and many other such applications are there. But more than research, why Resonance decided to request him to give this talk is that he is also a very good science popularizer, or rather he does a lot of videos. Almost 3 dozen videos in Malayalam are there in the YouTube where he tries to popularize chemistry, put some challenges for people in chemistry, and we could not find a better speaker to talk to young students here. So with these words, I request Sureshan to give his talk called A Click That Clicked Well. Sureshan.
Thank you, Professor Elias, for that introduction. And I also thank Team Resonance, especially Professors Suri and my colleague Mahesh, for having me as a speaker for this wonderful event. Actually, I was planning to give a generic talk of history, not science. And seeing a few lectures, I realized that I need to put some science also here. So I have put some science on the initial part of my talk. Then we will slowly go to history, but that history has some connection with chemistry. How some molecules dictated the history. So half of it will be more pedagogical, and the rest will be something of that connection, the connection between chemistry and history. Okay, this is the human population from AD 1 to 2013. In AD 1, we had about 25 crore population. I mean, our population was 25 crores. And it took about 1800 years to reach 1 billion mark, 100 crores. And next 1 billion reached in just 123 years. Then next one was in 32 years, then 15 years, then 13 years, 12 years. The last one was in 9 years. Now we are 8.265. This is today's population at 10:31 a.m., just after the first talk. And the interesting fact here is you see a sudden growth after 1900, a jump, right, in population. And also one interesting thing in today's population, you see this is birth today and this is death today. You can see that death is less than birth. It's not that we have increased our birth, it is we have decreased our death rate. That's why we could reach this population growth like this. So how we decrease the death or death rate is the question. And this is another one. This is average life expectancy of the globe actually. Again, till 2017 only I have the data. Currently the life expectancy, average life expectancy is 73 for the world. For India it is close to 70, 69 point something. So we were never above, or we never touched the world average for that matter. And if you see the life expectancy at 100 years ago, in 1920, it's about 30, 32, 33. That was the average life expectancy. Suddenly you can see the jump from here to 73. In India it was 24. 100 years ago it was 24 years, the life expectancy, average life expectancy. Now it is 70, as I said. So again, there is a growth from 1900 plus. So what has happened suddenly for this growth of population and also the growth of this life expectancy or the increase of this life expectancy? So this is the answer for that. Chemists have mastered the art of synthesis. So the chemists learn how to make molecules, specific molecules, pure molecules, and that has saved life. You can see this is the first organic molecule synthesized, that is urea. Wohler, right? Wohler synthesized urea from heating ammonium cyanide. And later so many molecules, these are some of the simplest molecules people have synthesized. We can make even much more complex molecule today. And these are the people who made these molecules. The names are there. Most of them are Nobel laureates, as you can see. And now again I am coming back to the life expectancy chart. So this is the first time Robert Koch said bacteria causes disease. Until now we did not know what causes disease actually. People believed that the wrath of goddess causes disease, for example in India. We didn't know, there is no germ. This is the first time a germ is causing a disease. The knowledge came only in 1900. The first antibiotic was an arsenic-based arsenamine. Then the second one was penicillin in 1928. Then streptomycin, vancomycin. These are all different antibiotics we introduced. And with the antibiotic, you can see the life expectancy also going. So that means we have reduced the death due to bacterial disease. This is not only because of this antibiotic. Today we have about more than 11,000 drug molecules, synthesized drug molecules for different diseases. And that is the one which saved the life and reduced the death rate. And this is not the only factor. There are other factors for the population growth, which is of course production of food material, which we'll come to later. Okay, olden days, the drug, our sources of drug used to be the natural material. So we isolate specific molecules and purify them and use them as drug molecules. Whether it is animal or plant material, these are different drugs actually. This is a broad spectrum cancer drug. This is a painkiller, morphine. And for example, this is of course, there are some, this is an anesthetic, but it has, of course, hallucinogen also, right? Cocaine is a hallucinogen, so it has other properties also. And this is, of course, this is penicillin, antibiotic. There are so many such which came from nature. And as I mentioned, today we can make any synthesis of any molecule of any complexity. For example, this is azadirachtin. Azadirachtin comes from Azadirachta indica, which is our neem tree, Indian neem tree. And this was synthesized by Stephen Lay from Cambridge University. And in the last, in this paper, he mentioned in the last paragraph, this has taken 22 years for him to synthesize, 40 postdocs, 40 PhD students. Finally, he synthesized 2 milligram. What is the use of that synthesis, right? You learn a lot of chemistry. That is one of the best use. Other than that, you cannot use such a material for any practical purpose, as a drug or something. So this is the time people started asking, we need something better. When properties are our goal, our properties are what? We need to use them as drug, for example. When properties are our goal, such complicated synthesis is not justified. We need a simple synthesis for using for a drug or any other purpose. So for example, this is a simple synthesis starting from phenol to paracetamol, right? First is nitration, you get 2 products, and also nitrophenol and paranitrophenol, and that you have to separate. Then the reduction, then acetylation. This is what we do. And we generate at every stage a lot of waste material which we don't want. For example, this we don't want in this particular step. And there are other byproducts during the reduction process. And we need to use high temperature and catalysts, activators, solvents, so many things. And this is the fact, actually. For every kilogram of a drug molecule, we make 100 kilogram of waste. So that's a reality. So that is the time this person, Barry Sharpless, he started thinking about something called click chemistry, a chemistry which will not give a byproduct, a harmful byproduct. If at all it gives a byproduct, it has to be benign byproduct like water. Such a simple chemistry should be there, and there should not be any harsh condition for that chemistry. And purification should be very easy. So such chemistry is called click chemistry, and one of the click chemistry is this one. If you have an azide, this is an organic azide, this is an organic alkyne, and if you heat them together, this is known chemistry by Rolf Huisgen in 1960s. And that's called Huisgen reaction. This is a dipolar cycloaddition reaction. You get a mixture of 2 products depending on the position of these attachments 1,4 and 1,5. So this is a good chemistry but useless chemistry because it gives a mixture of products. So no one has worried about this chemistry until recently when these guys started looking at adding a little bit of copper 1 to that. When you add copper 1, you don't need high temperature, it reacts at room temperature, and it only gives one of these products. So there is no purification required, in fact. So that is the click chemistry. So at very normal temperature, and you get almost quantitative yield for this particular chemistry. So essentially, it's like buckling up your belt, right? So clicking 2 entities and making a new molecule. So that's what it is, click chemistry, for which 3 of them got Nobel Prize, not this person. The Nobel Prize picture will come later. But these 2, Morton Meldal and Sharpless, did this reaction at the same time, actually. These are the 2 papers. You can see the submitted date. Only a few months between the submission dates, actually. So they simultaneously did without knowing that other one is doing. Actually, in his case, it was a serendipitous discovery. He was doing something else, and he stumbled upon that, whereas Sharpless was actually trying for such reactions. And anyway, both of them published almost at the same time, and both of them got Nobel Prize. And there was another person. So this is the reaction. Now, its application is a lot, actually, in medicine. This is the current day medicine discovery, how you develop a drug. First of all, you have to have a protein of your interest, or that's called target. And then you need to have a lot of molecules, usually in crores, actually. For example, ciprofloxacin came from 1 crore 25 lakhs molecules tested. 1 ciprofloxacin came out of that. So that is the number we are talking about. We need that many pure material, pure compounds. And then you need to develop an assay to read out whether this particular molecule is acting in favor of us or not. So that is called an assay. Then you identify a lead molecule of the 1 crore material, molecules. You identify about 100 molecules which are having some effect on that particular protein of interest. And that is called lead identification. Then you optimize this material to make better one, which is better activity, having better activity. Then you do the preclinical trials, usually on rodents and other organisms. Then do the clinical trials in humans, and phase 1 is of course on healthy individuals, and then phase 2 and phase 3. And finally to market, it will take about 15 years, and it takes about 12 to 15,000 crore rupees to bring out one drug to the market. This is usually the procedure. So what I want to say is you need molecules in large quantity, 1 crore kind of number. This is what we are talking about for screening. So click chemistry is very useful. For example, if you have this kind of azide and alkyne, you can have 5 alkanes together, 5 azide together, and you can mix them together. 5 different well plates actually, in well plates. And you can do individually, then you can have each well, you have 1 compound actually. Or you can do together also. You can treat all these 5 with 1 azide, then you have 5 molecules actually. And you can test that initially, then finally you can separate them and see which is the active ingredient. So if you have a 10-10 matrix, you are going to have 100 molecules in one reaction. If you have a matrix of 100 and 100, you are going to have 10,000 molecules in one reaction. So this is the power of this click chemistry, and which is helping the drug discovery a lot. These are some of the drugs already in the market or going to be in the market very soon, which is having this kind of the linkage, which is the triazole linkage, where I have marked in red. These are the linkages which is having, this is the triazole linkage. This is, for example, this is for epilepsy. This is an antibiotic. This is again a blood thinner. And these are for cancer. For different diseases, actually, we have this kind of triazole containing drugs already. Now people are developing vaccines. So for every cancer, there is a peculiar glycosylation. This is called N-acetyl galactose. This is one of the sugars we have. We have 9 different sugars in our body. So one sugar is, this particular one is called N-acetyl galactosamine. So this N-acetyl galactosamine will be too much expressed, or most of our protein will be glycosylated with this particular, in the case of cancer. So people develop this kind of antigen. You can make an antigen, right? So by hosting this kind of sugar molecules, a lot of sugar molecules as a possible antigen, and insert into the system so that antibody will be generated. So they are trying to make cancer vaccine, and this was in the late stage of clinical trials. So this is being done by Danishevsky. Using this, our click chemistry, you have a peptide having an alkane and an azide, and you can do the clicking, and very easy chemistry, you can make such antibodies. And also there are attempts to make antibody for, vaccine for HIV virus. Similar strategy. And in polymers, of course, polymers is, another area where click chemistry has a lot of possibilities and applications. You can have this kind of diazide and dialkane, you can have AB kind of polymers. You can have this kind of block of polymer. That means one polymer, another polymer, you can fix them together, you can have much better properties. So you can have a hydrophilic polymer, hydrophobic polymer, and you can tune the property by using this click chemistry. And also you have some azide in this particular polymer, and you can have a pendant unit with whatever pendant unit you want. Imagination is your limit, actually. So whatever you want, you can make, you can click them together. That is the beauty of, this is a brush polymer again. And in materials also, this is all our carbon nanotubes, graphene, and also fullerene. You can attach them because they are insoluble. To make them soluble or make them to have application, you can attach this kind of large polymers to this material using this simple click chemistry, essentially by introducing an alkyne, then clicking with azide. So now Bertozzi's work. So sometimes we may have to do this reaction inside the cell, inside a living organism. Then you cannot use copper 1 actually. Copper 1 is toxic. If you use copper 1, if you inject copper 1 to the cell will die. Why she wants to do this reaction? Because to track molecules. For example, she wants to study one of the proteins in the cell. If the whole auditorium is one cell and each of you are different proteins having different function, and if I want to study one of the protein, I want to tag you with something, a fluorescent material or so. So for tagging, this is very important. Usually biologists use GFP, right, which is a protein itself, which is having 238 amino acid, which is coming from Acuria victoria, which is a jellyfish. And that is huge actually. 238 amino acid, you are sticking to some another protein. So that make really heavy, and it cannot do the proper functioning actually. It cannot do the native functioning completely. So that's a problem. So in this, if you have something like this is clicking, you can directly click a very small fluorescent material to a protein. So that is the interest she has. So this is again non-chemistry. So she cannot put copper into the system. But this is already known. If you have a cyclooctane which is already strained, it is supposed to be SP, right? SP hybridized means it is supposed to be linear, and this is not linear. That means it is having a strain. To release the strain, the moment it sees an azide, it will react to form this kind of triazole. This is a very well-known chemistry actually, again in 60s. So she used this particular concept. So her idea is that somehow she want to introduce azide to the cell, then bring the probe, whatever the probe, fluorescent probe or drug molecule or targeting molecule, anything it can be, and with this alkane, then you introduce it to the cell, then it will automatically click, then you can see the cell glowing or the particular protein glowing. So this is the idea. So she has used the biosynthetic machinery actually. So as I said, there are 9 different sugars in our body. One is sialic acid. This is sialic acid. Another sugar is N-acetyl manosamine, and this is synthesized from N-acetyl manosamine. This is just an aldol reaction. So we have an enzyme called aldolase, and that aldolase make these two molecules. One is N-acetyl manosamine and pyruvic acid to do this aldol reaction and make the sialic acid. That's how sialic acid is synthesized in our body. So she used the same biosynthetic machinery. What she has done is, instead of acetyl, she has put an azide here. So it's just azidoacetyl mannose. Then she fed that azidoacetyl mannose to the cells, and cells started synthesizing this particular azidoacetyl sialic acid. And finally, this will be introduced to different proteins. Then whichever the protein is having this sialic acid, having an azide, she can visualize after putting an alkane with a fluorescent tag. This is the idea. So this is what she has done. After introducing the azide that way, she has put this biotin. Biotin is a ligand for streptavidin. Streptavidin is another protein, and biotin is a ligand for that, and very strong binding. And this streptavidin is already colored in the sense it is fluorescent. She made it fluorescent actually by FITC introduction. So now, if she introduces this one, it will click automatically here, and you can connect this with the fluorescent FITC avidin, and she can see the molecule of your interest. And this is the real picture image. Actually, you can see the cell membrane is glowing actually. So that means all the sialic acid is already on the cell membrane. All the proteins expressing sialic acid is in the cell membrane. And this is another chemistry she has done. This is with another sugar. This is called N-acetyl galactosamine. And she has put that to see the glycosylation pattern or N-acetyl galactylation, you say. So it is N-acetyl galactosamine. So at what stage this particular sugar is introducing into different proteins she wanted to study. So essentially she micro-injected this material, and that has become a donor actually within the system, and that transferred to different proteins to make mucin type glycosylations. And that she could visualize later by again introducing this kind of probe having this kind of coloration. Yeah, so they got Nobel Prize, 3 of them, Caroline Bertozzi, Sharpless, and Morten Meldal. Of course, for Sharpless, it was the 2nd Nobel Prize, who got 2 times in chemistry actually. Once it was for asymmetric epoxidation reaction. Anyway, now coming to my chemistry, how my chemistry is connected to this. So click, we are thinking about click without catalyst, which already Bertozzi has done. We don't even want solvent actually. So she has used solvent, right? So click without catalyst and solvent. Can you do click chemistry in the solid state is our question. So this is the idea. So you bring molecules like this. This is, take it as a monomer example actually. This is just a schematic. You have this complementary head and tail. That means once they are in proximity in the crystal lattice, they will automatically react. These are clickable material. This is azide and alkane for the time being. And if I crystallize them, the head and tail are at proximity. They are already at a very good distance, close distance. Then they can do reaction along this direction. You can get polymer of the same material in the crystal, within the crystal. And you can solve the structure of polymer by single crystal X-ray diffraction. Normally, polymer chemists make the polymers in solution. The moment you evaporate the solvent, what you get is an amorphous material. You don't get the crystal structure. But this way, you are synthesizing the polymer in the crystal state actually, and you can solve the structure by X-ray diffraction. So this is again, we started with sugar actually. This is a galactose derivative having an azide and alkane. I have shown this is the crystal structure. Azide and alkane are at proximity. And this is the packing diagram. It is forming in a head to tail fashion, actually chain in the molecule. This is the monomer. And if you keep the crystal for some time, 5 minutes is sufficient, it will automatically polymerize. You can see wherever the azide and alkane, that has become azide triazole actually. So it is a single crystal to single crystal transformation. You can solve the structure of this particular material. And we have done this for many, many other systems. These are some of the pictures, of the molecules we have seen. In every case, you can see there is a regio-specific reaction. Always we are getting the one which is getting the copper catalyzed one, right? One for selectivity. So this particular material is a hydrophobic polymer actually. You can coat surfaces with that polymer, then you can have hydrophobic surfaces actually. It has a lot of application for paints and other things. And this is again a sugar-like material, not sugar. This is a DNA actually. So we made DNA, homobasic DNA, using cytosine, and again in the crystal structure, again within the crystal, and you can solve the crystal structure by X-ray diffraction. And this is a different system, again squaramide. And this particular polymer from this one, actually, this is the structure of the polymer. And you can see these two dots. These dot means these are water from atmosphere. It is taking water from atmosphere. And you can release that water. That means you can get the water out just by blowing air at room temperature. At very low temperature, you can get the water out. Again, it will take water. In 6 minutes, you can do one cycle. And you can do several cycles per day. And you can get 18.5 liters of water per day per kilogram of this material from atmosphere. From where? Anywhere in the globe actually. Even in the Gulf countries where the relative humidity is much less. Even at 30% of relative humidity, this material can harvest water from atmosphere. Now, currently we don't have water. All of us don't have water. All of us in the globe. 10% of the global population don't have water now throughout the year. After 10 years, 40% of the global population will not have access to water. So that is a scenario. So people are looking for water from atmosphere. Actually, there is a lot of water in the atmosphere. 6 times water present in the oceans and earthly bodies. That much water is there in the atmosphere, in the air we breathe. So people are worrying about how to trap this kind of material. Usually people use MOF material. This year's Nobel Prize is for MOF, right, in chemistry. MOF is a metal organic framework. Metal has some connection or attraction with water molecules. It's a ligand. Water is a ligand for metal sometimes. And the problem is that coordination is much stronger. Much stronger means what? You need to put more energy to get the water out. Here it is a simple organic polymer. You can get this water out even at room temperature. But it's expensive as of now because this is made from amino acids. Anyway, so we have made a lot of such materials, so a lot of polymers. And of course, in some cases, you get the other selectivity also, but again, specific, only one selectivity. In this case, we are making helical polymers for various applications. Actually, these are having properties. You can use them as stationary phase for chromatographic separation. You can separate the enantiomers actually using this kind of material. And yeah, these are the ones. This is a ladder polymer. Again, crystal structure of the ladder polymer. You can see it's like a rope ladder, right? So this is one step, next step, like that. So it's going like that. So all kinds of structures you can make. And also using this same monomer, we can make 2 different polymers, having the difference in the regioselectivity. Only nature can do that so far. For example, nature can make, using glucose, cellulose, or starch, or 1,3-beta glucan. All these are starch polymers of glucose, same glucose. Only nature can do this. Similarly, isoprene is a monomer for rubber, natural rubber. Cis polyisoprene is natural rubber. And guttapercha is the trans form of that. So nature uses the same material to make different polymers. And we have shown that even chemists can do that. To make different polymers, you can use the same monomer chemistry, same monomer. So the next one is this one. This is very, very challenging for any polymer chemist to make a 2D polymer, right? 2D polymer, a sheet of polymer. You cannot make because once you put in solution, it randomly reacts and form a 3-dimensional network, right? Highly, not at all ordered material. You need this kind of sheet of polymers. This is possible only in crystals or on surfaces actually. If you can make a layer of molecules, monomer molecules aligned, then you can do this reaction. But crystallicity is possible. We started with this kind of monomers having 2 azide and 2 alkyne, and this is the crystal structure of those polymers, and this is actually the real crystal structures. Now what application you can think of from this one? And I can also change the cavity size. I have several such 2D polymers in the lab. So we can filter materials out of this one, actually different materials. So you can, for example, we are trying to desalinate water. Take the sea water and put it through this one, and you should get only pure water outside. So such filtration is possible. Nanofiltration is possible with such 2D material if you tune the cavity size. Yeah, this is the one example we have recently published. The same 2D material which is taking 2 water molecules and 1 water molecule, that is depending on the time. It is taken from atmosphere again, right? Just from the air. And it can also take methanol, it can take acetonitrile, and you can filter them, you can separate them. If you have a mixture of acetonitrile and methanol, you can selectively take out the methanol using this particular material. Yeah, this is a new reaction we have invented. This is called teak reaction. The previous one was topochemical azide alkane cycloaddition reaction. The copper catalyzer, the click chemistry for which Nobel Prize is called CUAC, right? CUAAC, copper catalyzed azide alkane cycloaddition reaction. We made it as topochemical. Topochemical means in the crystal. Topochemical azide alkane cycloaddition, TAC. Now, to rhyme with that, we made a teak also, topochemical enazide cycloaddition reaction, wherein only difference is instead of an alkane, you use an alkene, and what you get is a triazolene instead of a triazole. Triazole is aromatic, triazolene is not aromatic. That means you can kick away 1 molecule of nitrogen and you can get aziridine. So if I make this kind of backbone in my polymer or linkage in my polymer, I can make this to aziridine. Aziridine is highly reactive. You can open with any nucleophile. You have learned SN2 reaction with aziridine and oxiranes, right? So it can be opened with any nucleophile and you can have the backbone modified even after polymerization. So that's very wonderful. So some examples we have shown, we can even make chiral polymers. And an interesting thing is yesterday you heard, day for yesterday you heard Sudha's talk were origin of life, right? One of the unsolved mysteries in the origin of life is how the homochirality came. Only D sugars and L amino acid. That made life. If that homochirality is not there, there is no life. So how homochirality came from nowhere? Actually, no chirality to chirality is a big question. Right? These are some of the beliefs that it came from elsewhere, but from elsewhere is very difficult to believe because this comes with about 2600 to 800 degrees centigrade. And that temperature, no organic molecule will survive. No life will survive, no organic molecule will survive. So most probably it is terrestrial origin and it has happened here actually in the earth. How this homochirality happened is one of the unsolved challenges actually. So here we have an answer for that. So this is an achiral molecule. There is no chirality in that, right? This is a peptide, no chirality. But overall, that crystal is a chiral. So crystal can be chiral even if the molecule is not chiral. That is called supramolecular chirality. Now, if you trap that supramolecular chirality using a reaction, and you convert that to supramolecular chirality to molecular chirality, you are generating chiral molecule out of achiral material. This is called absolute asymmetric synthesis. So here we made an absolute asymmetric synthesis of a polymer. So interestingly, we have an ene reaction, right? Enazide reaction. In that reaction, it is generating a triazolene, which is having a chiral center. So we are generating a new chiral center. And because of the chirality of the overall chirality of the crystal, we are generating a new chiral center and a chirally pure material in this case actually. So we made a chiral polymer from an achiral material. And we are not saying that this is the reaction which formed the initial phase of Earth, right? No. We are saying that achiral monomers could have crystallized in the early Earth condition in chiral space group And that could have led to some reactions like photochemical reactions, and which could have led to chiral material, which could have catalyzed further chemistry and made chiral materials. That is same thing. Yeah, this is a half an hour, and I sort of finished my part of the chemistry part, right? So now I am talking about history, which I already promised. How molecules dictated history. Let us see how many I can see. I can stop at any time whenever the time is up, because there are several examples. So of course you know this, right? These are two different forms of carbon, right? Diamond and graphite. What is the difference between the two? Yeah, hardness is a property, right? What is molecularly how they are different? So let me take this is a brick, right? This brick can be packed like this, packed like this, packed like this, and n number of ways. And some of them are stable assembly, some of them are not, right? You know which one is stable, right? This will be more stable than any of those, right? So a material, this is an atom, for example. If it is an atom, it can be packed in different ways. That is called allotropes, right? So what we are saying in carbon is allotropes of carbon. One allotrope is a diamond, the other one is graphite, and there are other allotropes, right, like fullerenes, etc. And they have very different property. The packing dictates the property. And if it is a molecule, it is called polymorphs. If it is element, it is allotropes. And same meaning, polymorphs is for molecules. So that is what it is. Anyway, now coming to history. This is, of course, Napoleon. Napoleon used to be a very good ruler, actually. He had brought a lot of good things for France, especially Europe. Higher education, good tax code, road and sewage system. This was never existed there. And the most important thing is a uniform civil code. And Napoleon's civil code was used by the rest of the countries, actually making their own civil codes later, actually. Such is the civil code he made. Legalized divorce, then ended feudalism. A lot of good things he has done. But one mistake he has done is to attack Russia on some day. The day is 1812 with 600,000 army, 6 lakh army people. They marched to Russia in June. Now, when they reached this place, they reached here and coming back, when they reached this Bersina river, most of them died. Most of them died, only 6,000 people came back home. And that was the start of debacle of Napoleon Empire, actually. So they came in, by the time it was December, actually. What has happened? Their button, Napoleon's army's button is made up of tin, right? Tin buttons. Tin has a property that is called allotropes. This is what I told about allotropes, right? Tin has two forms of material. One is beta tin, which is the lustrous metallic tin. And it has an alpha form, actually. It's a gray tin. It is like ash-like powder. Okay. When the temperature goes below, for example, below 13 degrees centigrade, it will go to the other form. So in winter, in Russia, or especially in Russia, in December, it is very, very cold. And when it happened, I think I have a video for that. Yeah, this is the video. This is a metal piece of this particular tin getting cooled at the surface. You can see it is converting to the other form of the same tin, actually. So this is the alpha form of tin being formed. This is the beta form. So initially it was a metallic tin, and it became an ash-like tin, and they lost the button, actually, button of their coat. And they died. They were not holding the weapons, they were holding their coat, actually. And all of them were killed. And Napoleon did not know the chemistry, neither the people who designed this one. So this was one of these people consider this is one of the reasons Napoleon. Of course, later Battle of Waterloo is the one which he pathetically failed, and Napoleon Emperor was completely wiped out from Europe. But this is one of the reasons. Okay, so if this was not the case, Napoleon could have reached even India. And he would have made a lot of things, actually. He would have done a lot of good things, I would say. Anyway, next one is spices. So spices used to be very, very expensive stuff, right? Nutmeg, for example, used to be gold, much expensive than gold. those times. And this kind of yard workers in London used to get paid in clothes actually. If you do the head load work, you get 2 clothes actually. So that is the wages you get. So this is kind of the importance of this one. For example, Visigoths captured Roman Empire, right, in the AD 400 plus. So that time they asked ransom. What they have asked is only this one. Give us some pepper actually, just 3,000 pounds of pepper. We'll give you back your country. So this was the importance of spices that time. And how the spices reached Europe is a question. Spices are not there, right? Spices in the our part of the world, right? So it has reached through the Silk Road. This is the Silk Road from China. The silk goes from China, right? Silk goes from China to all the way to Europe. And this is a very, very difficult terrain, hilly terrain. Of course, you have desert, everything, and through the land, and it reaches here. Then finally, it goes to Venice. And Venice used to be the richest place actually. You used to read Merchants of Venice, right? It's a novel, right? So they used to be very rich because of the spice and silk. And the spice used to go from all the way from India to this through the Silk Route to reach here. And somehow these Venetians got this one, and they became very rich all the time. And this is the route. Later, this is the route after this years. Through the sea route actually, but through the shore, almost through the shore actually, small ships. Okay, so yeah, this is one of their favorite food actually, favorite spices actually. That time this was only produced in Kerala, this place actually. In the whole world, this was only here actually. Not anymore. Now the largest producer of pepper is Vietnam. India is in the 4th or 7th place. Okay, so they wanted to come to Kerala, definitely for Kerala, to reach, to get pepper. So Vasco da Gama, not only Vasco da Gama, many people try to reach here through the sea route. Sea route is much more easier than coming through this difficult Silk Route. Right, so they tried, these are different people tried from Portugal actually. Diego, they reached only up to here, from Lisbon to Cape Verde, then they died. Then next person is Dias. He reached up to Good Hope. This is Good Hope. Then again, he also died. Then later, Vasco da Gama somehow reached Calicut. Then he came to Cochin, then went to Goa and returned back. And after that, pepper came into the custody of Portuguese. Now, Portuguese became the richest people, not the Venetians anymore. This is the time Columbus told. He made a mistake though. He told Isabella and Ferdinand, said Earth is globular, so they have gone eastward. I will go westward to reach India. So my route will be shorter route. Give me some funding to make a ship. So he was funded. This is the first funding for science actually. And Isabella and Ferdinand gave some money, and he made a ship. And he made a calculation in his, the length was much more longer, right? He cannot reach India so easily going westward. But he reached America and he told this is India, right? West India, right? So the new world was invented because of this actually. Then later he made n number of visits to this American region. 4 voyages he has done, and he has controlled this area actually under Spanish control actually, especially this Caribbean islands and South America. And yeah, he later, they of course, the Spanish usually used to control Philippines also. They reached up to Philippines. They used to have all kinds of trade relation with this kind of countries here also, Indonesia, Philippines, etc. So that time Lisbon was part of Spain, right? Okay, now it is not. Now it is Portugal. Okay, the question is, what is in this material? It is the shape? No. It is the color? No. Why people are allured to that particular globular seed? It is because of this particular chemical inside, which is piperin. So in other words, this piperin has established and destroyed empires actually. And it has helped to find America, New World actually. Because Columbus also went for pepper only, but he did not find pepper because his calculation was wrong. The whole America was invented because of this one, and new movement has happened because of this particular material. This is piperin. This is nothing but piperin. Not only piperin, there are many other such chemicals, somehow it's not coming. There are other chemicals from spices actually there, those compounds. These are compounds which allude people, and that made the movement, and that made the development. Next is vitamin C. Vitamin C, of course. So in the 15th century, people started moving in bigger ships. Before that, it was only smaller ship through the shores, I mean close to the shores. Then they started making bigger ships and longer voyages. And they used to have a lot of cargoes, then large crew, because so many of them will die during the travel. So to accommodate that, they have extra people actually. Very limited space for sleeping, but they take more people because the value of a person is less actually, right? And this is their food. In long voyages, they cannot have fresh fruit or fresh food. So of course, this is salted beef or pork. And this is called hatak. Hatak means basically it's our maida mixed with the salt and water and baked actually, like a rock. And it's very difficult to eat. Sometimes this weevil, right? We will eat this one and make holes. So people usually, this is the same food for armies also. They like the weevil infested hatak than the normal hatak because it's easy to chew, right? It has pores and it will easily break. So this is the kind of food they used to eat in the ship during the voyages. In 5-6 weeks, they will get scurvy. And scurvy is such a bad state, people will die because you lose muscle so easily, so all the soft tissues will be lost. And especially you have this, the gums will be lost, teeth will be lost, and internal organs will be lost. This kind of patches will appear. And it's a so painful death actually. So basically our protein, or one particular protein called collagen synthesis is being affected because they don't have vitamin C. Vitamin C is required for collagen synthesis. They did not know what is the reason. So that was the reason, and they were missing collagen, and losing collagen is the death. So this is scurvy. For example, this is the first voyage, Vasco da Gama's visit to Kerala, right? He started with about 170 people. Only 54 of them survived actually. Because of, not COVID, because of this one, they died. Magellan, of course, you know, right, who circumvented the earth, right? It was in 1519. He circumvented the earth. That is not really true, right? We give the credit to him, but he was killed actually when he reached Philippines. He touched Philippines to have food actually. So that time the localites killed him. The rest of the crew came actually. 270 they started with 270, only 18 survived. 18 reached home back because of scurvy again. And these are called ghost ship. There is no one in the ship actually. Everyone died. This used to be very common scene actually in the sea. So many ghost ships used to be there. Everyone died during the passage, the voyage. About 2 million people, that means 20 lakhs people died out of this disease in the sea. And this is the first voyage of English East India Company to Good Hope actually, Cape Good Hope. He was the captain, Lancaster. He came with 4 ships. This is the Dragon is the flagship where he was there. In his ship, he has given lemon juices to people who is having this problem. He gave lemon juices. Only his people from his ship survived. Others died. That is the reason. And this is the voyage of Tasman. Tasman is a Dutch person. So he was moving to Australia to find they used to believe that there is some big land that side. This is called, they used to call it as Terra Australia Incognita, the unknown land of Australia. The meaning is that. So they want to find the unknown land of Australia. So Tasman went all the way through the Good Hope and this Madagascar, then this is Tasmania, right? After him, his name is, this is Tasmania, this is New Zealand. He did not touch there, he just saw New Zealand. He did not see Australia, though he made a circle of all these places, he did not see Australia. He went back. Only very few people reached home again. Most of his people also died because of scurvy. And this is the first time a RCT was done for scurvy. Actually, this is a Scottish naval surgeon, James Lind, and he was in this ship, and when 12 people got this scurvy, he made into 6 groups, and he did the clinical trial in the ship. This is the first clinical trial for that matter. He gave 2 people cider, actually, vinegar, basically vinegar form, right, from apple or something, the cider you make. Then 2 people got vinegar, and 2 of them got H2SO for unfortunate guys, sulfuric acid, then seawater, then some kind of a lemon tree, some kind of tea actually, decoction. And the last guys got 2 oranges and 1 lemon. And only they survived in few days. And this is the first RCT, and then they realized that, yeah, the lemon can save the lives. Even if it's known, why people are not taking it? This is a big question, right? Majority did not believe. Even in our case, it's like that, right? If we invent something, people won't believe, right? Majority did not believe. Then logistical problem, of course, was there, because you cannot bring citrus fruit into this one, right? It will die soon. I mean, it will perish. And growing citrus or ginger on the ship is very difficult, because you don't have such space, because every inch of that ship is very expensive. You need to transport material. These are for transporting material from back and forth. And something was very cheap actually, right, which is the human life. So what they have done is, instead of taking 100 people, they take 150, so that 50 let them die. This is what they used to do. Okay, this is a hero actually, James Cook. So he is actually hero in the sense he, at the very young age, he traveled to Canada, these Canadian islands, and he found those islands and claimed that island for British. And then later he was given this is the island actually. Later he was given a charge of finding this again terra incognita Australia. So he went to Australia. He went to Australia actually many number of times, actually 4 times. And his ship got wrecked into this one. This is the Great Barrier Reef. You heard of that, right? Around Australia, there is a barrier reef, which is actually rock like this one, right? Reefs actually. And his ship got collided, and it has got holes actually. This is about 15 to 150 kilometers from the shore. And people were manually removing the water from this one, and they were trying to do this one. They were really powerful, because he ensured that all of his team members should eat sauerkraut. Sauerkraut is cabbage, fermented cabbage, which is rich in vitamin C. And he gave vitamin C for all his crew members from day 1 actually. None of them died, and everyone were powerful, and the ship slid off, and he could reach Australia. Not only he could reach Australia, he reached 4 times, I mean, he made 4 times travel to Australia. This is his route actually. And sorry, 3 times actually. This is in 78, then 70, 72, 76. And first time he went to westward, then eastward, and he explored the whole area and claimed the land actually of Australia, New Zealand, Tasmania, everything for British. Okay, these all are his whatever he achieved. First circumnavigation of New Zealand, then first crossing of Atlantic Circle, then discovery of Great Barrier, then discovery of Hawaiian Islands. He was killed in Hawaii later, again the localites. So he was given, of course, he was an FRS Fellow of Royal Society, and he was also given the highest Copley Medal for his outstanding vision, and not losing any man in any of his travel. So yeah, in just 1 minute. So this is the structure of ascorbic acid. This is synthesized from glucose. We cannot synthesize. Many animals can synthesize, except primate and few organisms. Others can synthesize this one. This is the synthetic route actually by enzymatic route, and we cannot do, so we have to take from outside. Today we produce about 50,000 tons as a preservative. If you don't have this kind of preservative, we will have this kind of dangerous botulinum toxin, right, in food materials, which is really dangerous. So this is saving our life. And this is the last one. If vitamin C was not there, consider what would be the situation. Portuguese is the first to travel to India, right? They would have traveled further to Australia, and they would have claimed Australia. Portuguese would have been the language in Australia now. That is not the case. Magellan came, right, in 1521. He stopped Philippines because he did not have food. I mean, his people were dying. Stopped Philippines and he got killed. He could have come, if there was vitamin C, he knew knowledge of vitamin C, he could have reached the Spice Island, Indonesia, and he could have brought the control of spices actually, which later Spanish people, Dutch people claimed. So that would have changed the whole history, right? Tasman came here, he could have claimed Australia, New Zealand, then Dutch would have been the language of Australia, which never happened. The British actually started very, very late actually. If you see, they started only in 18th century. They would not have anything actually. If this vitamin C knowledge is there for Portuguese or Dutch or Spanish, they would have claimed all this land. British would not have had anything actually to claim. So the whole history would have changed if the vitamin C knowledge was known. So geography, so this is another one, so I will not go to that. I think this much only. Thank you very much.
Yeah, we have time for a few questions. Yeah.
Morning, sir. So it
Hello.
was an interesting lecture.
Thank you.
Sir, you told TEAC is done for alkenes.
Yes.
Can we do it for cyclic alkenes and alkynes? Like benzene and all?
No, benzene will not be reactive, right? In crystals, sometimes benzene is also reactive as a double bond, right? For example, this J.J. Vittal from National University of Singapore, he used to do 2+2 cycloaddition with benzene double bonds. So that is possible in crystal. If they are proximal enough, they will react, despite it is going from stability to instability actually, right? It is having aromatic stability. Still it can react if it is so close. The first question was what?
Like
Alkyne anyway reacts, of course. That's what the TAC reaction, right?
Okay.
The topochemical acid alkene side.
Hello, sir.
Yeah.
It really much isn't a question, but like you were mentioning chirality, I was reading something about mirror cells, but not on that scale, but just peptides with different chirality. Can you comment on how the future of medicine would look like given these new amino acids and we wouldn't have the enzymes to degrade them in our own body or something?
No, actually, the point is if this desymmetrization was not, the life would not have been. That is the issue. Now we cannot change the life, right? So now even if we make D-amino acid, of course we made D-amino acid much before that. Of course, that is being used as drugs also. Many D-amino acids are drug molecules because it cannot be chewed by peptidases or proteases actually. So there are cyclic peptides and even linear peptides which are drug molecules using D-amino acids. That's useful actually. So which cannot be recognized by our enzymes, right? Our enzymes can only recognize this as for chewing or cutting the hydrolysis reactions. So that's good actually.
Thank you, sir.
Yeah, question.
So very nice talk. Regarding the sudden spontaneous emergence of a chirality for the living systems, you argued that even if you didn't have a preferred chirality, but crystallization will give rise to a preferred chirality.
Yes.
But why one variety of chirality?
Exactly. So this is the question though. So there is something called Wiedmer ripening. Wiedmer ripening is, you are right, there can be equal proportion of both or chances, right? One time it can be D or it can be L. So Wiedmer ripening says that if they are in equilibrium, for example, the crystal and non-crystal version are in equilibrium, or in other words, it is dissolving and recrystallizing. So it can form to one of the forms. So that is called Wiedmer ripening. So that's already known. So one of the form can nucleate. Once it nucleates, the other form cannot grow because there is no other material actually. These are achiral material. This is not racemic material. So only one form will form. For example, there are people working on this. One Professor Otto is a Sibgen Otto. He works on this kind of system wherein he starts with an achiral material, then he stirs actually. The stirring will dissolve this material and one of them immediately nucleate and it grows only that form. So that's called Wiedmer ripening. It's there.
So does it mean that life originated in a very small pocket?
No.
Because if it was independent, you could have generated different chiralities.
Sure. So different parts of the world, it can be different chiralities, for sure. So we are not saying that it is the life's origin, but we are saying that chirality can be created like this, right? So that's what only we are saying, in a small pocket.
Okay, thank you.
Okay, any more question? Yeah, here, last question.
Sir, how seawater can be converted to drinking water, sir?
So if you have a method to filter out, sodium chloride is much more bigger in size than water molecule, right? So if you have a porous material or a filter, for example, in larger sense, which can permeate only water under gravity, not with pressure. So you take the membrane and put the seawater, and out of gravity, whatever drips down will be pure water. That is the idea. That's never successful so far. This is the idea actually.
Okay, sir.
But with pressure, it is known already.
Okay, so let us thank Professor Sureshan for an excellent presentation.
Thank you.
And on behalf of the organizers, a small moment.
Okay. Thank you. Very good. Thank you.
Thank you so much for an excellent talk, Professor Sureshan, and also Professor Anil Elias. We have maybe a 2-minute break, and I invite Professor Hema Somanathan to. It was supposed to be yesterday, and thank you for doing this. And I also invite Professor Nishant Nair to chair the session.
Yeah, good morning, everybody. And so today, I think the next lecture is going to be also an exciting one. Professor Hema Somnath is from IISER Trivandrum, and she's doing research on something very exciting on how plants and pollinators are interacting. And also, Professor Somnathan's lab name is also exciting. It's bee. I guess you're going to show what is the full form of bee. And I should also add that her students organized the morning walk today and going to be organizing tomorrow. So I think we should really thank her for helping us with that.
I hope I'm audible. A very good morning still to all of you, and I hope you're enjoying your stay as well as the sessions that the organizers have put together. I thank Resonance for this opportunity to speak to a lot of aspiring scientists, young scientists over here. And you know, this is something that whenever I get a chance, I don't miss this opportunity, because it's not just about talking about what I do, which I definitely enjoy, but it's also, you know, the questions that I get from an audience such as this, a very young audience, is often thought-provoking, and it helps me in my own, you know, research and things like that. So therefore, I never miss this opportunity. I'm sorry that I couldn't make it yesterday because of some personal circumstance. But yeah, so bear with me. So you people, you know, this is a campus which is like adjacent to a lot of very, very high, you know, endemism, biodiversity, and so on. We are in the southern tip of the Western Ghats, which is home to a lot of endemic flora, fauna, and so on. You will be amazed at, you know, the kind of life that thrives very close to where you are, including the big ones. right? We have all the venomous, the big four as they are called, king cobras to vipers and so on. You don't have to worry if you walk on the tarred roads, but just adjacent in the forest you find all of these. You are also very close to the Asian elephant, leopards, you name it, and we have it on the campus or adjacent to the campus. So, and my lab, I'm basically trained as an ecologist and an evolutionary biologist, so there's no better setting than this for me to sort of, do my work. So today what I'm going to do is to try and, share some of what we try to understand in a very simplified way. Please stop me if you have a pressing question, otherwise I can take questions at the end. So my lab, as was introduced here, is called the B Lab, and it's an acronym for Behavioral Ecology and Evolution Lab. And because we also study bees a lot, so therefore it became quite nice to call it the B Lab. So today what I'm going to do is, and I think all of you know that bees are important for pollinators, and we depend on pollinators for servicing crop fields as well as for keeping our forests to maintain diversity and so on. So therefore, I mean, this is something that I think is very elementary and everybody appreciates. It becomes very, for us, for researchers like me, it also becomes very easy to do fundamental research using bees, because they also have this, the applied value of bees in food security and so on. So a lot of, just to introduce you to pollination as an ecosystem service, a large majority of angiosperm plants actually depend on bees for pollination. They depend on other pollinators, but bees are specifically important. So by some estimates, about 90% of flowering plants depend on one animal or the other for pollinators, and 75% of our food crops, cocoa, coffee, you name it, I mean lots of vegetable crops, cucurbits and so on, tomatoes, lady's finger, all of these things depend on animals for pollination. And about 35% of the global crop productivity depends on animal pollinators. Now, so therefore, bees and other pollinators become very important in terms of food security, in terms of biodiversity, in terms of poverty elimination and so on. Now, when you come to bees as pollinators, they are considered to be super pollinators for several reasons. One is because they are very versatile, they visit a huge variety of crops as well as other plants, and they also travel large distances and they can perform pollination as a service. So they are very important, and in India we don't have a really good estimate about how valuable bees are, but we do know that they contribute a lot to pollination services. But in the US, where they are very, very good at record keeping of this nature, one of the estimates is that bee pollination contributes about $14 billion per annum to crop production in the US. Now, when we talk about bees, we are used to thinking about honeybees. Yes, honeybees are very important, but there are thousands of other bees that are not social, but they live by themselves. Okay, and these are what we call as the solitary bees, and their role is often underappreciated, right? So, and as I said, we don't have very good estimates for the tropics, simply because we don't keep records very well, and we don't really understand how valuable are various kinds of pollinators to many of our crop plants. Now here is a list of some of these food plants that are serviced by bees, and you will recognize that many of these are actually temperate plants, right? So, but there are, I mean, in the Indian scenario, all our pulses, like your dals of various kinds, are all pollinated by one bee or the other. Now, so keeping this in mind, the United Nations has sort of declared that there is a day which is called the World Bee Day, and that happens to fall on May 20th. And the reason why bees are so important for pollination is simply because they are so critically dependent on pollinators for food, for themselves. Bees don't care about pollinating plants, but they care about collecting food for themselves, right? And for the brood. They are collecting, if you think about social bees or even solitary bees, Social bees especially, because their colonies tend to be really large, thousands of individuals, and so therefore they have to collect food not just for themselves or a handful of individuals, but for the entire colony. So therefore, they become very important because they are searching for, nectar is the reward that they get, that plants provide to bees, and pollen as well as nectar is what the bees sort of take back. In the process of taking pollen out of flowers, they also contribute to pollination. Now, they are called central place foragers simply because they have a central place which is called the home, right? And they have to carry all of this, what they collect from flowers, back home. And so, which means that they should also not just find flowers, okay, but they should be able to return back home, for which they have to remember where they went and where they are in relation to home, yeah. And we will see in a moment how they do this. So, people have been interested in bees for a very long time because of the food value, which is the honey that they produce, right. People, I mean, we did not particularly know about pollination, but if you look at even very old rock paintings such as this is an example of a rock painting in Ganesh Ghati in central India, where you can see that these are Apis dorsata colonies, and you can see that the honey is being harvested. There is another example of another rock painting which is also depicts the same thing. And an interesting thing you can see over here is all of these paintings from this area actually depicts women who have climbed on top of these trees and cliffs in order to harvest honey. Okay. Now, that is a lost skill. We do not see women doing this anymore. Honey collecting, honey gathering, honey harvesting is mainly done by men. Yeah, this is something that has been of interest to me too. Now, we began to recognize the importance of bees as pollinators after the work of this gentleman called Sprengel, who was very interested in the diversity of angiosperm flowers. Okay. Why do flowers look so different? Different colors, different shapes, patterns, smells, and so on. And so, he began to systematically document this diversity of flowers, and later on this gentleman, whom you all know very well, right, who is he? Charles Darwin, of course. He was specially interested, he was interested in a number of things, and he had a lot to say about a number of things, but he was specifically interested in orchids. Okay. And he used to wonder about why are orchids so complex in their shape and structure and so on. And in orchids, what happens is that the reward, which is the pollen, is often hidden so well inside the flower that it becomes very difficult for a pollinator to access it. So, this was a puzzle, because if a bee has to be rewarded and it has to learn and it has to come back to a flower, then you should not make it so hard for it to find the reward, find its food, right. So, this was something which Darwin used to think about, and he had hypothesized a lot of things about why orchids are so complex in structure, shape, and so on. And subsequently today we know a lot about orchid biology, but this also started with Darwin. The other interesting thing, especially when we think about honeybees, is that they are considered to be the pinnacle of social evolution, right. So, the interesting thing about honeybees is that what you see here is a big colony of giant honeybees, which is called Apis dorsata, and you can see here that their colonies consist of thousands of individuals, literally. These individuals are organized, a colony is organized into castes. Okay. So, there is only one reproductive individual who is called the queen, and most of the other individuals are workers. There are a few males, the workers are all females. There are a few males in the colony, but never more than 5 to 10% which are males. Okay. But so, essentially it is a matriarchal society with a lot of thousands of females doing a lot of tasks within the colony, right. So, and what kinds of tasks do they perform? You have the queen, which is a reproductive individual, she is the only reproductive individual. Then you have the males, which are also called drones, and they do the job of reproduction. They never reproduce within the colony, they do not mate with their sisters or mother, they go out and then they are mating with other virgin queens and so on. That is a different story for a different time. And you have the rest of the colony is all workers who are females, And these females do all the other tasks within the colony. Now, what are these tasks that they do? When a worker is very young, she does tasks within the colony. Okay? She does tasks like cleaning, maintaining hygiene, feeding the larvae, taking care of the eggs, throwing out the dead bodies because they want to keep the colony clean, disease free and so on. So they go around inspecting the comb, various cells in the comb, and then they check if everything is okay, and things like that. And as they become older, so then they begin to become foragers, which is when they leave the hive, they go search for flowers, bring back pollen and nectar, and then they store it in the cells and so on. So but that happens much later in their lifespan. So and then there are some which do this job of guarding the hive. Okay, so because a hive consists of lots of food, okay, which is very attractive to birds, to other insects and so on, and the larvae are rich sources of protein, and so then the entire colony has to be defended from intruders, predators, parasites and so on. And so there are guard bees such as what you see over here, waiting at the entrance. They look like commandos ready to launch an attack. So these are the guard bees. These are workers who are building new combs, and the foragers can look for, sometimes if it's a very hot day, they can also search for water and bring back water. And what you can see here is some bees drinking water, collecting water, and this is their predator, which is a hornet, it's a wasp. And here what you can see is a forager who is collecting pollen from flowers. Now, I was telling you that they are critically dependent on flowers, and they need to travel long distances sometimes to find flowers. And so there were these nice experiments, very simple, very elegant, doesn't require a 5 crore machine, but gave a lot of very valuable insights into insect learning, animal cognition, a lot of animal neurobiology and so on, insect neurobiology and so on. So bees have been valuable not just for their role as pollinators, but also because they gave very valuable insights into learning, memory, cognition, physiology and so on in terms of in insects. So other than Drosophila, after Drosophila, bees are probably most well studied amongst insects. So what you can see here are these experiments which these researchers performed with the western honeybee in Europe, where the colony you can see, it consists of, these are the hives that were placed in a landscape, and then each bee was marked. Okay, so you know the identity of each bee, and then they look at how far do these bees go. Okay, so they place nectar sources, and then they try to record where the bees go. And you can see here that sometimes they can go travel as far as 10 kilometers. They don't do this on a daily basis. On a daily basis, it's about a kilometer that a bee can, will fly in order to find flowers and come back. But if there are lean seasons, or if the demand for food in the colony is very, very high, those times they can even travel 10 to 12 kilometers in search of food. Now, if they are traveling so far, that means they have to come back reliably. And so how do they do this has been something that has been fascinating to scientists for quite some time. Now, this gentleman here is Carl von Frisch, who won the Nobel Prize in 1973 for some very, very remarkable discoveries. Okay? One is he showed that bees are capable of color vision. Okay? Till that time, we thought that only humans were capable of color vision, and everybody else saw the world in black and white or grayscales. But today we know that insects are capable of color vision. They see floral colors very vividly. They see colors because they have a UV blue and green receptors. They even see colors that we don't see, right? So there are patterns in flowers that have co-evolved with bees, and these patterns are visible to bees, and they help them to land, to orient to flowers, and perform various such functions. Now, the other remarkable discovery made by von Frisch is is the bee dance, right, or the waggle dance. How many of you have heard of the bee dance or the waggle dance? Figure of 8 dance, it's called the waggle dance, figure of 8 dance. So not too many. So let me take a moment to explain what it is. I also have a video of it. Now what happens is that I was telling you that a honeybee colony consists of tens and thousands of individuals. Now if each of these individuals have to go out and find food, okay, and they need lots of food because there are thousands of hungry larvae in the colony, and so they have to be very efficient at this. Now if each of them had to go out into the landscape, search for resources on their own, right, that doesn't seem to be very efficient way of feeding thousands of individuals. It would probably work if you have to feed tens of individuals, but not when you have to feed thousands of individuals. So therefore what do in order to improve efficiency? They have something which is called a symbolic language. We have, now I am talking to you, and because everybody here understands English, I am able to communicate even with the back row, the person in the back row. Now so therefore I can get this message across to about 100 people in this audience over here very effectively with a mic and English as the language of communication. I can get my message across. Now they don't have a language in the sense that we think of language, right, but they have something which is called the symbolic language or the waggle dance. Now I will tell you what a waggle dance is. A waggle dance is a way of communicating to your hive mates where a particular food source is located. So therefore the other foragers in the colony don't have to travel on their own searching for food. Instead they can use the information that a successful forager has provided in order to go to that food source. So therefore that becomes a very efficient way of collecting food in large quantities for the colony. Now, yeah, so the work of this man and spurred a lot of interest in honeybees as models for not just pollination but also physiology, navigation and so on. Sensory biology, vision and things like that. So now we do know that they travel these large distances and they come back home reliably because they are using the sun as a compass to guide them. Any point wherever they are, they use the sun as a compass to come back home reliably. Yeah. We also know that they can use any landmarks like trees, distant hills. They can learn these features of the land, of the landscape, pretty much like how we do. When you go into a city, okay, you remember buildings, you remember some statue, you remember some, these kinds of features in order to find your way to a favorite restaurant of yours, right. Similar to that, the bees can also do that. They learn whatever they can learn. They will learn, artificial structures like buildings, they will learn billboards, they will learn anything that they can use in order to come back home or to find flowers and get back home. At a flower, they can learn various features of a flower. Okay, they can learn the smell, they can learn the color, just like how you sit here and imagine a rose. Okay, just close your eyes, you know what a rose looks like, you know what it smells like, you can, right, you can sense it, right. Similar to that, they can remember these features that are that a flower presents. Now, and this is the thing which I was saying, that they also have this symbolic language. So these were remarkable discoveries. I won't get into each of these, but I will tell you, I won't talk about the color vision part over here. That itself is a very, very interesting story with respect to how did he actually discover and do very elegant experiments to actually say that they have color vision and it is different from intensity based vision, which is black and what we call as black and white vision or grayscale vision, right. So in humans we know that when the light levels drop, we become poor with color vision beyond a point, and then we revert to black and white vision, right. So we can only see, we can't discern colors very well. Now similar to that, you have to actually tell, when you do these experiments, you have to say whether a bee is actually seeing color or is it seeing the grayscale property of of an object, right. So that is something which he did using these very, very elegant experiments, which I will not talk about, but it was a remarkable discovery. Now with respect to the waggle dance, okay. Now when a bee finds food and it's successful in finding food. It comes back home and then it performs on the comb surface, it performs a particular dance, okay, which is what we call as the waggle dance, which tells the other bees that are watching this bee, okay, they are called the followers. They are surrounding this bee and they are watching this bee, and they tell this bee where is a particular food source in relation to the position of the sun, okay. So, directional information, and how far is this food source from the hive and how attractive it is. So, all of these in these three kinds of information are coded into this waggle dance, which makes it possible for the followers to actually go in that direction, fly that distance and find those flowers. So, I will skip this. So, therefore, today we know that bees use many cues in order to locate flowers. Okay. Initially, once color vision was discovered, people thought that color was the most important cue. Yes, it is a very fundamental cue, but today we know that they can learn various features of flowers like size, contrast with the background. So, the contrast that the flower produces with respect to the leafy background, they can learn odors, they can learn patterns, subtle patterns like this on flowers, they can learn the texture of a flower, they can learn shapes, they can even sense temperature of flowers to make a decision whether to land on a flower or not, and they can sense electric fields on flowers which tells them whether a flower is still receptive, is producing rewards and so on. And all of this information can be used by bees in order to find flowers. It is not that they used all of these for every flower, but they are capable of using all of these different cues in order to locate flowers. And the other part of this story is what people have been excited about is to try and understand how do bees actually forage efficiently. So, one is they have something which is called an innate template, which is sort of like a hardwired preference, okay, for a particular feature of a flower. Okay. Today we know that they have some color biases for certain kinds of colors. Even a bee that has never seen a flower before, so a newborn bee in the laboratory, when you expose them to colors, an array of colors, you will see that they show a preference for mostly short wavelength colors. And though they can learn other colors, when a bee has never seen color before and has never experienced a flower before, so we call them as naive bees. These naive bees will show a preference for certain or a bias for certain kinds of colors over others. They also show some bias for symmetry versus asymmetry. So, there are various kinds of biases that inexperienced bees will show, and this is what we call as innate templates. Yeah? They will also learn very quickly. Okay? Like I said, they have a bias for short wavelength colors, but with just one or two trials, if you can expose them to, or you can give them an opportunity to learn that a long wavelength color like red or orange is rewarding, right? You can find, if you tell them and teach them that there is nectar there, they will quickly switch their preferences. So, this is something that they are also very adaptable and they can quickly switch preferences, which is a very useful behavior for survival. When you think about nature, there are flowers of different kinds, and when one flower stops flowering, they have to switch to the next. So, they can learn this very quickly, and so that this is another way in which they forage efficiently, right. Now, they also remember. Okay. So, if you give them 5 different kinds of flowers, and some of them are more rewarding, they have maybe more concentrated sucrose, the others have less concentrated sucrose, they will quickly learn which is more concentrated, which contains higher rewards, and so they would attach greater value to those over others. And you can teach them these. The way we do this in the laboratory is that, say, we can associate blue with higher sucrose content and yellow with low sucrose content. They will show a preference for blue. If you switch it, then they will start, and now the yellow becomes more rewarding. So, then they will immediately learn that in 1 or 2 trials and switch to yellow. So this is something which makes them explorative in the field, which is very important for survival, and adaptable too. Now, the other kind of cue that they use is social information, right? So you as individuals here, you don't have to sample every restaurant yourself in order to decide whether, you know, the food is good, whether the dosa is good or not. You will rely on what your friends tell you. You will rely on social media, you know, likes and so on, and you will use all of that information in order to make a decision whether to go to that restaurant or not, right? So similar to that, bees can also use social information. What kinds of social information do they use? One is these waggle dance. What is a waggling, dancing bee telling? Is it an attractive resource? Is it far or near? You know, all of these are used in the decision making process, and that is the way they use social information. Now, we also learn from each other in a different way. We learn by observation, right? There is a task being performed. We observe someone who is performing that task and we try to imitate that, and we learn how to do something, right? If a baby or if a young child learns how to uncap a bottle, it's by observation or trial and error, and then the learning happens. So similar to that, bees are known to watch other bees, okay, watch what other bees are doing, okay, and see the preferences of other bees, and then learn by observation too. So that's also a kind of social information that they use in order to forage very efficiently. Let me talk about a little bit about the waggle dance part. So an important thing for you to remember is that you know what is the size of a bee, okay? It's just a few milligrams in weight. So you can imagine the head of a bee is really tiny, the brain of a bee is even tinier, and if you want to know what is the exact size of the brain of a bee, you just take a very fine ballpoint and put a dot on a paper. That is the size of the brain of a bee. So it's a very, very tiny brain that we are talking about. It has thousands of neurons, but even with that, you can see that they are capable of learning quite complex features. They can remember what is an attractive flower, what flower to avoid, which has toxic nectar or which has sweet nectar, where is a particular resource present. All of these things are things that they can learn, remember, and store into their memory and retrieve when it is required. Now here what we have is a bee that is performing a waggle dance on the surface of the comb, right? So she has found food, it's attractive, so she comes back home and she performs this waggle dance. Now what she does is that she waggles her abdomen from side to side, okay, and then makes a return loop to the same starting position, and then again waggles her abdomen from side to side and comes back in a return loop, okay, and does this in an alternative fashion like this, and this creates sort of like a figure of 8 kind of a shape, okay, and at the same time there are a number of recruits or followers that are observing her dance, okay. Now what this dance tells is the direction of the food in relation to the position of the sun and the distance to the food, okay. Now the waggle run, the length of the waggle run or the duration of the waggle run codes for the distance to the food source. Now here you can see there are 3 examples, okay, of bees waggling, and you can see that this is the position of the sun, the hive is here, the food is here, okay. So you can see that this is a bee that is waggling, okay. She goes from 1, 2, 3 back, 4, then 5, 6, 7, 8 back, right. So this is the waggle dance. And if the food were present in this case, in this third case, you can see that the food is present at an angle of 60 degrees to the sun, okay. So you can see that she orients the dance, okay. So the sun is vertically overhead, and she makes an angle of 60 degrees in relation to the sun, which tells the followers that food is present at an angle of 60 degrees to the position of the sun. Yeah. So this is the way in which bees can convey information, which makes it efficient for followers to go out and search for food. Now you might ask me this question as to how precise is the dance? Do they get to the exact spot? it's not that precise, of course. There is a degree of error, there is a small margin of error, but it takes you in that direction and it takes you roughly where it is, because distance information as well as directional information is coded. Okay, so therefore, it gets you as close to the resource, and then there it's a local search which you might do yourself, or you might find other bees also arriving there, and then it becomes much more easier, and you can optimize the search for food in this manner. Now, and this is all important because you, as I show you here, you can see that bees are trying to collect food for a large number of individuals, and they also have to come back with food, not just for themselves, but also to feed. As you can see, each of these cells, you can see the larvae inside each of these cells. So they have to go into these cells and feed the larvae. They have to feed the queen also, because the queen never leaves the hive, right? Once she is mated, then her job is only lay eggs, hundreds of eggs day after day. She lives much longer. A queen bee lives about 3 or 4 years, depending on the species. Workers are much short-lived. They live for a couple of weeks, in some species a couple of months. So therefore, they have a much shorter lifespan, and they have to feed the queen bee in order to sustain the colony, so that she keeps on laying eggs till she is dead. What you can see is a grab from one of our own videos over here, and you can see here that this is the dancer, and you can see a number of bees surrounding her, trying to watch this dancing bee, and you can see that on her legs, you can see that this white mass on the legs is pollen that she has collected from flowers, right? Now I'll try to show you a video, hopefully it will work.
Honeybees have some fascinating abilities, among them being able to communicate by performing a unique dance. It informs hive mates where a newly discovered food source is located. Every cycle of this waggle dance roughly describes the shape of the figure 8. Let's rewind and look in more detail. The bee only waggles on a part of its route, the straight run, indicated here by the waved line. The secret lies in the direction of the straight run, or to be more precise, in the angle between the straight run and the perpendicular, which in this case is 90 degrees to the left. This tells the other bees that food is available 90 degrees to the left of the sun. If the angle is 60 degrees to the right, they'll be flying 60 degrees to the right of the sun.
Yeah. So when this symbolic dance was sort of being studied, people were trying to understand. So I told you what a dance codes for, but let me tell you how people arrived at that now. Okay. So there are a number of steps that are involved in a dance. So there is the measurement of direction. Okay. So a bee has to know in what direction she has flown, and she has to convey that information properly to the hive mates. She has to measure the distance that she has flown. Okay, has she gone 500 meters, 1.2 kilometers, 3 kilometers, and so on, because she has to convey it accurately to the other bees. She has to code that. Okay, so now she has a measurement of direction and distance. Now that has to be coded into the dance, and she also has to code distance into the dance, and finally she has to transfer all of this information to the bees that are observing her dance. So today what we know is that in terms of measurement of distance, they will use the position of the sun, and as we know, the sun travels across the sky. Okay, the position of the sun is not sort of the same. At 12 noon it's right above, but then in the morning and in the evening the sun is more towards the horizon. So they can use a time compensated sun compass in order to tell bees and include that in their dance. When all the dances are essentially performed in relation to the position of the sun, so that makes it more accurate for a follower bee to get that information. They will also use in their navigation, they will use polarized pattern of light. As we know that the light from the sun is polarized when it reaches the atmosphere, and create certain patterns that we are not able to see, but many insects are able to see this polarized pattern of sunlight, and they can use that pattern in order to find their way back home. And as I said before, they will use any kinds of landmarks that they can use in order to find their way back, because you must understand that from a bee's perspective, it's very important that she returns back home without getting lost. It's not like another species which does not build a shelter or does not have, to feed other individuals, where they can rest anywhere and take shelter anywhere temporarily, but a bee has to come back home with very, very high fidelity. So, this was again demonstrated by Carl von Frisch, that there is a time compensated sun compass which allows navigation properly in a landscape. So, next comes to the measurement of distance. How do they actually measure? We have a smartwatch which tells you how many steps you took, or we have a compass which tells you, we have a, one of those devices which tells you, a GPS device which tells you how far you walked, in what direction, and so on. So, there were two hypotheses that were proposed. The first one is that they must know how far they have flown simply by how much energy they have spent. Okay? We feel that when we walk 5 kilometers, we are more tired, so we have a sense of, oh, if I walk 5 kilometers, it takes this much of time and I feel more tired. And if I walk only 2 kilometers, then I don't feel as tired and it takes only much lesser time. So, we have a sense of all of this that we integrate in order to try and understand how far we are from home, right. So, similar to that, so one of the hypotheses was that they have some way, we don't understand which way, how they do it, that is a different question, but they must be having some way of measuring the amount of energy that they have spent, okay, in order to know how far they have gone. The second is called the optic flow hypothesis, okay, which I will come to you in a moment. And so, these were two competing hypotheses that were tested in order to try and understand how do bees actually measure distance. So, there are very interesting experiments, and this it's also to give you a flavor of how these experiments can be done, and sort of like out of the box thinking, where you may or may not succeed, but you try various ways in order to answer these questions. So, in order to test the energy expenditure hypothesis, what researchers did is that they did a bunch of experiments where they they caught bees, they attached weights to the bees. Okay. So, they had two sets of bees, one without weights attached to them and one with weights attached to them, lead weights. Okay. And then they made them fly, okay. And then what would you expect is that if a bee is carrying heavy weights, so then she would probably feel that she has flown much longer distance, whereas a normal bee without these weights will signal that she would be more accurate in her estimate. So, by using these kinds of experiments and by making bees fly in normal conditions as well as very windy conditions, okay, making the flight more challenging under windy conditions, a bee that's flying against the wind is going to signal that she has flown longer distance as compared to a bee that is flying in sort of steady conditions. Then they make bees fly uphill or downhill. Okay. Uphill, of course, we know is more difficult, we feel more fatigued, right. So, the same thing for a bee as compared to downhill searches. Okay. Then headwind versus tailwind, walking searches, they make bees walk and then estimate distance. So, all of these bunch of experiments were done in order to test the energy expenditure hypothesis, which essentially is asking whether they measure distance flown using some internal measure of how much energy is spent, okay, giving them two conditions, one which is more energy expending and the other which is less or is a normal condition or a control condition, and then looking at how accurately they can predict how far they have gone. Now, all of these experiments are done using artificial feeders where you know where the feeder is placed. So, you as an experimenter, you know the distance to the feeder. Okay. So, and you train the bees to go there first, and before you do these experiments, weighing them down, making them go uphill, downhill and all of that. So therefore, you can control the distance and you know how accurately a bee is giving information back through the waggle dances. Okay. So they come back, they perform the waggle dances, and a lead weighted bee, for example, is going to signal that it went much further than it actually went, right. So these are nice, very simple but challenging experiments to do, not as simple as I am making it out to be, but interesting novel ideas in order to test this hypothesis. The other is the optic flow hypothesis. Now, what does optic flow basically mean, or what it tells is that a honeybee, you know, when you travel in a car, right, the environment is streaming across your retina, right. When you are sitting in a bus or a train, the environment is streaming in your retina. So essentially, the bee has some way, again we do not know what way, okay, but some way of sensing how much of optic flow or how much of visual information streams across its retina in order to know how far it has gone. Yeah. So this was the second hypothesis, which is called the optic flow hypothesis. Again, very nice experiments, very ingenious experiments were performed, where they had bees fly over two different scenes, as you can see A and B over here. Now A is a cluttered environment, okay, there is a lot of optic flow that is going to be generated in this environment, okay, whereas B, which is flying over still water, right, is going to generate more or less optic flow on the retina. So imagine a bee flying over water versus a bee flying through that environment, which is crowded with trees. So a bee is going to signal greater distances here and lesser distances here. So this was what was done by, this was what was tested by making them fly through environments which provided greater optic flow or through environments that provided lesser optic flow. Again, very nice experiments where they had bees trained from the hive to enter a long tunnel such as this. You can see the bees flying through the tunnel. Each bee was marked, so you know the identity of each bee. Okay. And the way we do these experiments is we either color mark them with paints, okay, waterproof paints, or these days we use number tags which the sensors can read and so on. And then we film the whole thing. We use high speed cameras which can sort of film the flight of the bee. So here you can see a bee flying through this tunnel, and you can see that this is a checkered tunnel. Now the idea here is that the checkers provide optic flow information. It's like a cluttered environment, right? So that's exactly what these experimenters wanted to do, that they wanted to provide them with a cluttered environment, right? They know the length of this tunnel, and why do bees behave and do these experiments and participate in these experiments is simply because at the end of the tunnel they will find food. You can motivate them with food and they will do almost anything for you with food, okay? Just like humans, not very different, right? So once they have learned, okay, to fly through this tunnel, then the optic flow is provided. So otherwise it's a plain tunnel, okay. They learn if you fly this distance, at the end of the tunnel there is food. So they will go, and they will go back and tell others to go, right, using the waggle dance. So nicely then you can test these bees. So once they are ready, once they are trained, then you provide whatever, you can manipulate how much of optic flow you want to provide, right. You can increase the amount of optic flow, you can decrease the amount of optic flow by changing the checker size, the black and white, this thing and all of that. And then you can see when they come back, when they go, they feed, okay, and they come back and then they dance, then they are going to estimate the distance, okay. They would have estimated the distance, and if they are using optic flow as a cue, then they will tell others how, how far they have flown through the tunnel. People also did these experiments outdoors. So the first experiment that I showed you was in a lab situation, right. So there are limitations to how long a tunnel you can have. You can have a tunnel which is like maybe 20 meters, at the max, was some of them even shorter. But if you do these experiments outdoors, so then you can really increase the distances and the complexity of these experiments. So here was an experiment that was done in Australia, where this is the location of the hive, and they trained one set of bees to fly over land, okay, to a feeder that is placed over here, and another set of bees to fly over land and water and land. So, you can see here the distance is the same, okay, that both these sets of bees have to fly, but here they have to also fly over water. So, about 1/3 of the distance is over water. Now, the expectation here is that these bees that fly only over land will experience greater optic flow, okay, and these bees that they should estimate the distance accurately, whereas these bees that fly over water and land, okay, will experience lesser optic flow, and they should what they signal when they come back in the form of their dances will show greater error. This is the expectation. Am I with you? Yeah? Yeah. So, now what we know is that let me just tell you the third step also before I give you other details. Now, so the third step is that after they have measured direction, after they have measured distance, they have to code it into the, I'll finish, code it into their dances. And today we know that they code it into their dances by sort of using gravity, landmarks and so on in order to be able to tell distances accurately. I think I've sort of covered this slide, so I will leave it at that. And let me also tell you then, not every forager sort of comes back and dances. There are foragers which will go out, if the food is not attractive, then they will not dance. Okay? So, what determines whether a bee dances? There are multiple factors that determine the motivation to dance, and that is it depends on the distance to the patch, it depends on the handling time, how complex the flower is, how difficult it is, the quality of the resources that are present, all of this. So, therefore, the resource value will determine whether a bee will dance or not, and the need of the colony, okay, whether there are lots of bees that are hungry and there is starvation, or whether there is very little food in the colony, all of that also will influence the decision to dance or not to dance, right. So, then once a bee dances, then there is the flight vector which the follower bees are looking at, and the same flight vector is then taken by the follower bees in order to locate the food. Now, of these two competing hypotheses that I told you, we currently know that the energy expenditure hypothesis, there is not much support, or there is no support for the energy expenditure hypothesis, but the optic flow hypothesis is something that is well supported. Lots of other studies on other honeybee species have shown that this is kind of a conserved mechanism in order to determine the flight direction. And today we use this information, or we use this in order to test how far bees go in various kinds of landscapes. Now, we know that with habitat modification, like plantations, agro-ecosystems, cities, urban areas, forests getting fragmented and so on, there is an interest in trying to understand how do bees fare in these different landscapes, right? How far do they go, what kinds of forage are they finding and so on. So, therefore, we do these experiments in different kinds of landscapes, and let me give you some of these. Again, the steps are the same, where we sort of have these 4 different landscapes which are available in the vicinity of the campus. So, in the city and in the paddy fields and in forests and in rubber plantations, where these hives are placed in all of these different landscapes, and then we allow bees to find feeders that we place, the artificial feeders, and then when they come back and dance, then we look at what is the information that is contained in the dance, and then we plot them something like this. Okay. So, for example, in the rubber plantations, you can see that each of these dots represents a distance that is signaled by the bee, when she comes back. And you can see here that the median distance is about 400, or let us say less than 500 meters. In the rice fields, the median distance is about less than 300 meters. In the forest, they are traveling also a shorter distance of about 300 meters, and in the city landscapes, they are again traveling about this. So, all of this, I mean, this is not, this is very little preliminary data, but what this tells you is that we can estimate using the dance as a tool to try and understand how do bees actually fare in different kinds of landscapes. So, with that I will end my talk and not get into bees that are active at low light levels. Yeah. Thank you.
Thank you so much for the amazing talk. I'm sure there are questions from students.
Hello, ma'am. It was an interesting session. My question is, from a physics perspective, how significant are electric field interaction in bee flower pollination behavior?
That's a great question, and we don't have the answer yet, because the fact that bees can use electric fields was discovered about 6 years ago, to be very precise. And we know that they can sense electric fields around flowers, okay, in order to make a decision as to whether to land on a flower or not. But electric cues, as you know, I mean, these are like very local electric cues, and even if it does come into the picture in decision making, it's at the very, very final stages of making a decision. Okay, so imagine a bee that's flying out of her hive hundreds of meters away. Okay, there are other cues that become important first, and when she is very close to the flower, in order to a bunch of flowers over here, whether to visit the white one or the red one, that will depend on electric cues, because freshly opened flowers have produced a stronger field as compared to flowers that are already depleted by other pollinators or are past the fertile phase and so on. So, they might use it in order to distinguish between flowers at that very local scale. Yeah.
Thank you, ma'am.
Hello, ma'am. Hello, ma'am. So, how do bees convey that the color contrast and the quality of the food in the dance, or they don't?
Color they do not convey through the dance. Okay. So, color is something which every bee experiences individually. Okay. So, they can, what I was trying to say is that they can learn colors, okay, and they can learn a range. So, they have these UV blue and green receptors, okay, which means that they can see a bunch of colors which we can't see using the UV receptors. But color is not information that is passed through the dance. It's not socially transmitted.
And food quality?
Food quality is what I was saying is that if a food is very attractive, let's say it's very, very high in sucrose, 30%, 40% sucrose content. So then what they do is that I told you that waggle phase, when she's shaking her abdomen and moving, they will perform it with greater tempo. When dancers get excited, they are dancing very fast, or when the music is very upbeat, right? So similar to that, the tempo of the dance changes. So that informs the bees that are watching that the food is very attractive.
And ma'am, you showed a slide where in different location, like in rubber and rubber forest, and in city they travel different distances, but all the distances were almost same. So
yeah, so that
how the optics,
this slide, optical, right?
Yes.
Here, over here. So this is actually very preliminary analysis. As you can see that the sample size of the bees is very low over here, right? But I can tell you I did not have time to integrate all of that because we are still to make graphs and figures for the new data. But what we can see finally is that in forests they are traveling much lower distances in order to find resources. The average forager is traveling much closer in forests as compared to, so forest and city are comparable. Again, city because they find lots of ornamental avenue plants which are planted at close distances and all of that. But in rubber and in rice fields, they have to travel much further in order to find resources. So therefore, rubber and rice fields are not very good environments for the field, for the bee to find flowers, whereas city and forest were sort of comparable. Yeah. In terms of distance, okay? In terms of quality and of the resource, because the reward is not just about sugar, but it's also about amino acids and various other things, so that the forest fares better. Yeah.
Okay, next question.
Good afternoon, ma'am. Ma'am, you explained how the waggle dance conveys information about the direction and the food quality, but how will they convey distance?
Distance, right?
Yeah.
Yeah. So the length of that waggle run, right? The straight run gives information. So I told you that they make the straight run where they're waggling the abdomen from side to side. So that the length to which the length of that straight run gives information about the distance to the food. So at some point they go and then they turn around and they come back, and then again the straight run, and then there's the return run, right? So that the length of that straight run gives information about
So
distance.
are there any like limits or constrictions on the distance that they can like convey? Like, is there like a specific range?
So let me, what I couldn't talk about is that this distance thing is quite conserved across species of honeybees and across habitats. Okay. So basically you do a study in a forest near Trivandrum and a forest near Bangalore, okay, it's more or less the same with a very, very small margin of error. And also there are interesting experiments where they got two species to live in one hive, okay, they can even learn each other's dance. Yeah. So that tells you that evolutionarily speaking, these are quite conserved.
Thank you, ma'am.
Any more questions?
Just two things actually. One is you talked about the error in the direction indication. Is it correlated with the altitude of the sun? The other question is, when you speak about that dance, you said this length, then the tempo. So when you have a sine wave like that, there will be amplitude, wavelength. So the waggle will be there for some period of time only, or the whole, does that differ?
The first question was about the sun, the altitude of the sun.
No, you spoke about the error in direction.
Error in direction.
It seemed
yeah.
curious because sun is overhead, you don't have any direction.
So in fact, that's an excellent question because they avoid flying at 12 noon.
Only later in the day,
yeah.
earlier.
They avoid flying at 12 noon because of precisely this issue.
Then also little
they
altitude.
also avoid flying when the sun is blocked temporarily, when the cloud comes across or something. You know, they will immediately sit on some tree or some branch wherever, okay, because they don't want to get lost, right? So they will just, so we did these experiments where we also catch the bees and we drive in a car, okay, like 10 kilometers away, and then we ask at what distance do they all get lost, okay, so we get an estimate. So of course, you know, when at greater distances, if we block all the cues, we are keeping them in a box, okay, and we are releasing them under darker conditions, many of them don't return home. So they absolutely need the sun, and they will avoid so it's a very good question. The second question was about the tempo and the direction.
The things which can vary.
The amplitude and things, right? So basically what they do is that they keep the tempo. So let's say it's a very attractive resource, so then the tempo is very fast, okay. The length of that
tempo fast means the frequency
the abdomen waving
is larger. If you're thinking of it as a sine wave in
so
it's a sine wave,
let me tell you in biologist terms, okay, they move the abdomen from side to side, okay, so it's a side to side motion which they make as they are moving forward like this. So that the tempo of that, okay, it can be slow or it can be this. So that seems to convey some information. The length that they travel in that long, the straight run also varies with direction.
Thank
you. Sorry, distance. Yeah.
Thank you. I think maybe we can all give a big round of applause to Professor Somanathan for really amazing talk. We have a small token of appreciation from the Resonance team.
Thank you. We had an excellent 3 talks today, and we have 2 more to look forward to. I thank Professor Nishant Nair also to have shared this session. So let's go for lunch and be back here before 2:30. Let's be on time. Thank you.